Good morning everybody and welcome to Dive 417 with ROV SuBastian. We are here on the eastern side of Ashmore Reef on the northwest shelf of Australia, and today we are exploring the Mesophotic zone of the Ashmore Marine Park. We're going to be diving at around between 150 and 100 meters depth today, and as our other dives we will be starting out running transects and then later in the dive having a look around and collecting biodiversity samples. I'm Dr. Karen Miller. I'm from the Australian Institute of Marine Science. I'm the chief scientist here on board the RV Falkor for the Australian Mesophotic Coral Ecosystems voyage, and along with Narrator Wilson from the Western Australian Museum, we'll be taking you through the dive today. We're just started on the first of our transects. We're at 150 meters depth. As you can see, lots of sand here today, some lovely sculptured sand waves, and plenty of activity in the water column early this morning. So we're just going to be gradually making our way along the seafloor for around 300 meters, collecting imagery that we'll use to process back in the lab to quantify the habitats and the benthic biodiversity. So it looks like it's going to be a nice relaxing start to the morning, so I hope you enjoy diving with us today. Looks like we didn't get our morning nautilus this morning.
No.
Although we did get our morning sea snake as we were descending.
Someone left the porch light on.
Oh, that's a flying fish, is it? Oh, he's just slipped out of the screen. Oh, here he comes. There's probably a much more technical word for them. Oh, he's kind of cute.
Yeah.
Not quite a flying fish, but a similar principle.
I think they call robin fish. Looks like a
What are
robin.
they called, sorry?
Might be called robin fish.
Robin fish.
Possibly.
Okay. We'll have to wait for a fish expert to let us know what they're called. They're very cute.
Triglidae.
That's quite a nice looking shell there, look at that.
Gurnard.
Take a photo of that shell, will you?
Form fish. They get their
It looks
name
very
from
dead,
one
but
sentence.
Yes,
Do you
John
want
Lodge.
DOI or?
can we just mark that, because Narrator might be keen to
Yeah,
come back and
I
have a look
certainly
at it.
We're on it right now. Mark, Adam's gonna do it. Good size.
It is.
Yeah. Mark, waypoint on that, mark that.
As we travel along the transects, the idea is to record continuous imagery and try not to stop too often. So what we do is we take waypoints when we see something of interest, and once we finish the transect we're able to make our way back and stop and look at things in much more detail. Yes, there is another one of those fish. Oh, the lasers aren't on, I just
Lasers.
realised. Not that there's a lot to measure yet this morning. There's something very satisfying about sand waves, but typically we don't find very many things living around them because the sediment is so dynamic. It's shifting around all the time due to typically a lot of current action, and so you very rarely see communities of things like sea fans or sponges associated with these areas. Wow, there's a huge amount of plankton in the water today. Lots of larval fish and squid, all sorts of things buzzing around there. Actually making it quite difficult to see the seafloor. They certainly are attracted to the lights on the ROV. Whoa, that I think made it worse.
So I think if we can adjust these. The problem is that the brighter the lights, you should go to that, but it's not working. We just hit a massive field of,
Yeah, oh wow.
see if we can,
It's a good job there isn't anything on the
yeah.
on the seabed to see.
See if I can shift them up a little bit, bright lights. This is a freezing amount of krill.
I feel like it's following us.
I believe they are.
They're having a real party in the lights of the ROV, that's for sure.
But there's food though, there's other things that might come and eat it.
That's a good point.
Well, I was thinking more like a whale shark, yeah.
And they did in fact see a whale
They
shark
did?
a
Oh,
couple of
big
days
one,
ago
yeah.
up on the reef.
Look at this, it's like a complete
Oh yeah,
band. It's interesting.
that's
the way it's
phenomenal.
formed
We're
up.
just looking at the plankton in another camera view, and it's just phenomenal. Oh, you can see that there now. It's phenomenal.
It's like a hurricane of food. Yeah.
It would definitely be a good place to be if you were a whale shark.
Later. Later. Excellent.
I think there may be a sea urchin on the sand, but can't quite tell.
Find out. Discuss it. Okay.
That is a phenomenal amount of
Yeah, it's a
zooplankton.
lot.
Let's hope it goes away when the sun comes up.
Trying to get
Awesome.
slipping it back. Right.
Before the show?
Yeah.
Really is just as well there is only sand on the seafloor because it's literally obliterated by this plankton. The krill, you see the cuttlefish, the squid, sorry, juvenile fish, all sorts of things in there.
Shark. Shark.
Yeah.
Oh, and there is a shark there. Not a filter feeding one though.
Oh,
We've just spotted a shark cruising behind us.
he's not interested. Do you know
No, he's decided we're not worth investigating any further and is swimming away.
might find your water samples might have a few extra visitors when we get there.
water in there.
Yes, it'll just be correct.
Oh
won't it?
no. Now you'll flush them twice.
Yes, you'll need a coarse filter.
Think of all those extra samples you're going to get.
I think I might.
What we could do is sit down, kill the lights for a wee bit, and then
That's a good
take
idea.
the samples, and
Yeah.
then maybe it'll
Sounds great.
clear up a little bit.
It's loaded.
Just keep it moving, doesn't have to be long. Right, RV, just to let you know, we're going to be stopping the water sample shortly, but we don't want you to stop. Just continue to your waypoint.
Copy that, Russ. Continue to waypoint.
Okay, that's its midpoint, so I'm going to basically come here, sit down, kill the lights. Just for a second or two.
So we're just stopping now to take some water samples, but because of all the krill, we're turning the lights off, hoping that they will disappear for a minute. So yeah, if everything's gone black at your end, don't panic. The feed hasn't stopped, we're just hoping the krill will disappear. The water samples that we're taking is for environmental DNA analysis. On board Falkor, we've got a great crew of scientists from a range of different organisations, and we each have our own scientific objectives. We have an AIMS at UWA student on board, and he is collecting water samples for the environmental DNA analysis, and the idea is that we will process those samples to see if the DNA that we see in the water column is the same or reflects the benthic biodiversity that we see on the seafloor. So, all that helped a bit. So we're just going to trigger 3 of the Niskin bottles, which are designed to collect water. You might see them trigger off in the background, but we use the ROV arm to activate the switch so that they collect water samples.
That's good.
There goes the first one.
Good.
Second one.
Good.
And the third one.
Check. Water samples 1, 2, and 3 complete.
Excellent.
looks like there's not much in it too, so hopefully there's not too many
Not too much krill.
extra bits.
Once we get those water samples back on board, they'll be filtered to collect the DNA that's in them, although I think today we might do a coarse filter to get the krill out first. And then later back in the lab they'll be processed and all the DNA inside the sample will be
Yeah, ready to
sequenced.
continue transects.
So we'll continue on now with our transect, just slowly moving along the seafloor, taking the imagery. About another 150 metres to go.
ROV. Do you want to go eat?
Can we go this way?
Yes, ROV, go ahead.
Or actually, I'll
Are you
go
able
first.
to,
and then I'll go
Are
grab
you able
something.
to pop up just a little bit?
Cruise is on.
Yeah.
Okay, and
Cheers,
off
thanks,
dead.
Jason.
I just finished the water samples and heading up. We're back.
What are we doing? Just laterally to starboard. Lateral to starboard? Yeah. I'm just gonna push it, just get a little bit ahead of the ship.
I'll mark that sea star as a waypoint. We'll come back and see if it's different to the ones we've seen before. How are you this morning, Ben?
Just peach.
Good.
Hard to see anything now, can it?
I know, it's quite difficult with all that plankton, isn't it? Luckily there's not much on the seafloor to see. Has the sun come up outside yet?
Yes, it's
It
starting
has?
to.
Okay.
It was so beautiful. It was like the sunrise. I don't like reading stories like that. Submariners.
I guess tea. Cheers.
Another sea star. Can we mark that as a waypoint?
Sure. Slowly, slowly. Okay.
Oh, a sponge with a cry note on it. Excitement. Actually, something down there. Looks like we're just transitioning into a slightly different seabed type. You can see it's gone from lovely sand waves to a sort of more rubbly coarse sand with some larger lumps in it. We were expecting that based on our multi-beam data, particularly the backscatter, which gives us a sense of how hard the seafloor is, and we could see a transition between the southern part of our transect and the northern part of the transect, which now we're starting to understand what the difference in the backscatter was telling us between the sand and this sort of slightly more consolidated area. Hopefully we'll see a few more things living in this part of the transect where hopefully there's actually something to attach to and live on.
Look how thick it is, by the way.
Oh my goodness.
That's how thick it is. You can see on this camera.
That's crazy, yeah.
You can see the corner.
Fish. Center Street.
Cuttlefish.
Yeah.
Oh,
Right here.
See.
You know that.
that sea star looks very familiar.
Yeah, we've done that.
It does look like another stick out the back there, doesn't it? There is a small cay on Ashmore Reef, of which there are a reasonable number of trees growing on it. We have periodically seen the odd bit of tree on the seafloor.
Bridge RV, updated waypoint.
I would expect that to have washed off the island.
Sorry, Rusty, was that update the waypoint?
That's correct.
Starting to see a few sponges coming in now.
There's a sea started.
Nice and what's the sea star? Oh, off your road. Yes, there is crawling along the.
Like the plume. See that there?
Yeah.
If your feet was going to smell bad, it comes up.
We might have time to give it a good rinse before we get back up.
It's not going to be happy.
There will be krill in every corner.
It's happened before where we had a couple of animals that got stuck and then it was baking in the sun
Ooh.
and it wasn't very nice. We couldn't find it, it was right inside the frame.
Oh no.
There was something in there and it was like, took quite a long time to get rid of that smell.
You've caught nautilus on the frame. Come back, there's a nautilus on it.
Oh, really? Wow.
We stuck between the cables.
Oh,
We threw it back
no.
though.
Yeah, it's alive.
Did it survive the journey? Did you throw it back in and then swim away?
Yeah, it's too beautiful too.
Oh, yeah. Good morning, narrator. Yep. Oh no, this isn't very much at all. Oh yeah. At one stage we couldn't see the seabed. We're just waiting for the whale shark to come cruising past, right in front of the camera, preferably.
I was distracted up yesterday to try to get some compression on something.
And it's like something like that. Yeah, like your picture over there. Yeah, I just pull it down.
Must have put light in because you can't notice the limits of teeth.
Your eyes, it's not too much.
I wish.
Good, I think. Did the lasers go away or can I not see them again? They're often hard to see against this background. Oh, there they are.
Yeah. 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 Just anything. Most of them work out to like that was like 2 seconds. Yeah.
Very blue today, or turquoise. Aquamarine. Took a lot of blue.
That was just for the actual talk. Not back work. Because you guys finished that work at 10. Yeah. Yeah. Yeah.
Not very far away now.
That works.
If you want to, yeah.
I enjoy doing that printing in 6 hours and see what it does.
Maybe I should not hold enough to be something. Give me a second.
Should I have to over to you. We're just coming up to the end of the transect now. There's a couple of systems that we have a look at them.
How far apart is this?
Not, you can see there, not that far, so we're at the northern end of transect. And then it's a bit shallow.
Over to the
Yeah, so I don't know how much we're looking at. There's
all the time.
so this is the second half of the transect has been this sort of consolidated stuff, but the first half was this really sandy stuff. So I don't know whether you want to look through that. It's not usually very much within the sample. It's too mobile. So yeah, I'll leave it up to you as to how much you want on this. Probably good to have a little bit of a close look. Yeah. I'm going to leave you with narrator. It's 7:30, time for our regular morning meeting at the bridge. So I'll hand over to narrator to guide you through the rest of the dive. Well, the rest of the this transect anyway. Action. Oh, stuff.
Morning.
Morning, madam.
Morning, everyone.
How's narrator this morning?
Yeah, pretty good, thanks. Yep. How about you?
It's peachy.
Peachy, that's excellent.
We should get out the krill here. Good on. Sorry. That'd be funny if you did that. OG krill. Oh, that's so hard to do that. It's a feature of yeah. Right, should we get a picture up?
Wow. Krill ball is a bit out of control.
Okay, we've got trusty flights in 122 meters.
Control works.
Yeah, go ahead.
Yeah, just for information, Russ, I'm bringing the head, ship's head more towards the southeast.
Okay, that's fine. Yeah, thanks a lot.
No worries.
Not bad. No, that's definitely 6. Kilometer 6, yeah.
Oh, I think it deserves more than a 6. Look at that.
Oh no, you haven't seen it earlier on. You couldn't see it.
Really? Oh,
Yeah.
God.
Oh, I've moved up to 7 now. 8. I can't hear it. 8, yeah.
Okay, that seems fair.
It is 7. So you got 4 meters to go. One past about a couple of meters, which we started shooting shy. next one. Is that, yeah, that's the one there.
Oh, look at that. It's getting thicker.
Oh, it's been like that pretty much the whole run. Okay, that's past our end of transect there.
Let's start the cruise.
Yeah, let's just have a look at some
Right, Jeremy,
rocky
we're going to
bits
start heading
somewhere.
west now. Is this your final heading for the moment?
Negative, that's kind of a fair bit round stuff, maybe another 10-20 degrees.
Okay. If I just give you a waypoint heading west, you can just sort of make your way there whenever you're available.
Yeah, yeah, that's fine. No problem. Just move waypoint. I'll try and do it then.
Sure, we'll just cruise over. Thanks a lot.
Perfect.
Yeah, these look great. Good.
We'll have a bit of time anyway because we have to let the ship change its head and then move so we can
Ah, perfect.
scout around here. Do you want to look in here a bit?
Yeah.
There it is.
It's all yours. Sorry.
That's right. I'm just going to have a little close look at the seafloor here. It kind of seems not to host so much life as some of the areas, but there's always life. So I'm just going to take the opportunity to have a close look and also let the ship reposition.
control direction.
Yeah, go ahead.
Yeah, this heading's looking pretty good, and yeah, you can see I'm heading over to the
Yep.
waypoint. Are you going to want to bring me a little bit further south? If
We'll
I'm on
be.
this heading?
We're just going to be stopping briefly. You just keep moving at your speed you're on. We'll just. I'll knock you down as we get across there.
Okay, understood.
So I'll probably move like the waypoint 50 meters south of that position, so about there.
Okay.
And then that'll bring us into that top. I'm going to start off at it's basically the northerly point, and then work our way south.
Got it. Okay, so you want me to head to that position? Is that right?
Yeah, that'd be great.
Okay, will do.
General direction. Thanks a lot.
Shell top right.
The homie.
No, top right is an empty shell.
Oh, yeah. Whoops. Another homie.
What is it? Yeah.
Wow, it is busy. Busy, busy, busy down there.
What's it? Hermes?
I'm not eligible.
Can't get in now.
It's not eligible yet. It's only 65 an hour. I can't be able to do it. Our age group won't be available in Canada until September, October.
Oops. Sorry. Let us continue on. Um,
shell.
It's coming for us. Seeing us off.
So
Is that with the colour? So you probably know actually.
yeah, I knew where it was on the drawing. I showed him it.
Can we stop in front of this one here?
Yeah.
Thank you.
Control groups, are you happy with my current speed?
Yes, that should be good. Oh, we're going to be stopping here in a moment.
So, do you want me to stop?
Yeah, you might as well, Jason. We might be here for a few more minutes.
Okay.
Reg RV, can we all stop in the ship move, please?
Yeah, you're stopping.
Okay.
Yeah, Russ, we're already stopped.
Roger, I'll just check it, thanks. So the plan was just to, once we get the vehicle, push codes, right, find out how many push codes for the manufacturer.
Do they want?
Yep. I want them.
We've got
40.
actually, there's a couple around. Sorry, for the slide with us. I might hold him down, and so you're gonna start heading up towards the 100 bar. Yep. Okay. So let's do this. Did you talk to Marty?
Yep.
He's coming down south.
Yeah.
Or is he coming there?
He's going he's not ready.
Stop. Okay.
He's not
Do you want me to bring? I'm gonna bring him around to here then. How's that?
Yeah, sounds good.
Hey, Jason, Adam here. That's an updated waypoint
Updated waypoint.
Oh, you're sampling it?
No, we're looking.
Yeah, I'll just bring it down so we're off the back. Right now we're like, he's out left field. Should be right. 100 meters. Anyways, if you're looking at it, just get it stuff. Change the with that. Yeah, he's probably gonna even more start.
That's fine. He went subside, he was like 90. Was heading east.
Yeah.
Turned back going 45.
Yeah, well, he's gonna keep coming started with it.
Thank you. Just finished my gum thing.
No, no.
Just until he comes around again.
So yeah, 40 push course, no bioboxes is doable.
They want any of those core covers or any of those?
Yeah, but it's looking at the space. We regularly got enough to put those push course back wheels again.
We've got the 2 block holders, so that's 16 right there. And then another 24 on the front, so that's 3 racks of 8. 3 racks of 8 in the middle. And then we put a couple more.
We need the tubes and bungs and stuff.
Big anemones here.
Stop here.
So you won't have the tools.
Just with the crystals, he basically took an inventory of all of those.
Where's the tubes?
I don't know, but
You should be up there.
maybe we should stop and look at the star.
Which one is it?
And now two.
See that part? Never seen it. It gets close.
Just the way the camera's facing, or is it actually low?
Thank you.
Bridge, RV.
Go ahead.
Hey Jason, just wondering what kind of speed can you make in that heading?
Probably pushed it up a little bit, but yeah, maybe 0.3.
Okay. Yeah, we're gonna transect over to the 100 mark. She's looking at about 400 meters or so, but just let us know how it works for you. Cheers, thanks. Jason, that's an updated waypoint.
Okay.
Thank you.
No, I couldn't go in there because it's still in there. No, I did,
There
yeah.
should be a pencil there, but right? No pencil.
Yeah. Before the.
Seagull benchmarks.
Oh.
Yeah. Okay. It's good. Yeah. The fly. Run the downhill run. Yeah. We get a couple more special
You
goodies.
can probably go a tiny bit faster.
Roger.
It's not so exciting.
We'll finish our breakfast.
Bridge control.
Hello, Ben.
Hey, Luke. Yeah, we could go a little bit faster. Oh, okay, never mind.
Well, yeah, we're trying.
Roger that.
At the moment, we're kind of fighting the carry, so we're going as quick as we can.
Roger that.
Take a rod to the right and just have a look around and let them get my feet.
So it looks like this is one of those benthic ctenophores that we've been seeing. We'll try and pop down close so we can have a good look and probably collect it.
Okay, great.
Yeah, go ahead, Luke.
Yeah, just slightly bring my heading around to starboard as we do this move, so maybe 5 to 10 degrees.
Yeah, copy that. Yeah, I've just seen the current changing there too, so
Yeah,
that's fine.
thanks.
Thank you. So for the size, it looks quiver size, so that's nice too. Yep.
It's quiver sized.
Excellent. Always good check.
One that was ballerina pointed.
It's lovely to see it.
There's colour somewhere in between some imagery.
That's right.
The pinky coloured one,
Yep.
but with the violet edge.
That's right. We had some that were one that was red and one. Yeah, it's great.
So you know that one. It's the best one. All that there. No.
Cheers.
Ben, would we be able to hop a tiny bit closer to it? I think we'll have to collect it and also just to get some more close-up imagery. Thank you. Hopefully we'll be able to zoom in on a bit more fine detail here. This is one of those beautiful benthic ctenophores we've been finding.
Actually increase the shutter speed a bit.
What's open?
Try and get you better color.
Ah, cheers.
They're not a focus. Is that better? That's pretty good.
Sorry, didn't mean to do quite that harsh. Awesome. If we can go ahead and collect this guy, it'd be fantastic. I think he's quiver sized. Yeah.
Watching out.
This is great to be able to get a final specimen of this species on our last deep dive of the trip. Well, I think the numbers are all mixed up from our...
We look at the numbers and everything before we close.
Pardon?
I think we get the numbers also if they match.
Yeah, that's fine. We'll fix it. Okay.
Okay, watching it.
No, these three are all mixed up. Beautiful.
Watching out. One stop.
Thank you very much.
That's not descriptive. That's different to the descriptive.
It is. It is. I'm interested in this. Are we able just to hop a bit closer to that urchin? I mean, it looks like it might have a little thing on the top.
Yeah, sure.
Yes, indeed. So this urchin is carrying one of those eulimid parasites that I was looking for. This is fantastic. Beautiful big animal. You can normally these parasites can be quite small, so it's nice to see one that just actually looks like a snail riding on top. And there's possibly some other little hitchhikers as well. You can see some other shapes in amongst the spine, so this will be a fantastic specimen to add to our collection. Did you get a thank you? So that will be both urchin and obviously limit, but you could just leave it with urchin.
That's right, I'll put it on there.
Okay, cool. Awesome.
So,
Somehow avoiding the little tiny snail on top.
Challenging with that.
Snail on top. Yeah, that's it. That white thing on top?
That's right, yep. And there's a little one. Oh, it's about to go under a rock. Sense shade.
Is it too fast?
Awesome. Beautiful job.
Watching out.
Uh-oh. He's moving.
Charlie.
They're so slippery, aren't they? They're so good
Could we
at
just
moving.
pause there for a sec? Luke, sorry.
yeah, buzzing here.
Thank you.
Thanks.
I can run for it.
Yep.
It's amazing how fast they can move.
I know. I guess they have a lot of legs.
True.
A lot of feet. Feet, yeah. Wow.
He's wise to us now.
It's going back the other way. Get away. It's climbing up.
It's a bit of crawl on. Which is perfect.
Slime around.
And your position.
Just try this. Is there a rock there?
Huh?
Yeah.
Actually, no rock. Just do one, so. Here.
Yeah, high damage. It's like it's a photo. Slippery.
I think it sort of pushes itself big and small and if it's quicker to fling it into a bio box, also feel free to do that.
It's just a cunning little fellow. Position. Gentle like a baby.
Fabulous. Thank you so much for that.
And this is 23 here.
Yes.
That's good. Alright.
Great. Thank you.
Morning, Chloe.
Good morning, how are you guys?
Very good. Very good.
Maybe a column underneath.
Oh, yeah. Alright, happy to move on, thank you.
We haven't made it very far this morning from the end of our 150 metre transect towards our 100 metre transect. Just sort of, there's a little bit of distance between those 2 points, and we're just having a little look along the way and collecting some samples.
Updated waypoint, thank you.
Thanks, Luke. Need something?
Oh, let's just stop there though. Oh no. No, sorry, continue. Just a whole,
Carry on.
such a trade-off. Oh, that's an interesting star. Yeah, can we stop and look at that star though? Sorry. Or maybe it's a, yeah.
Oh, thing I just picked up.
I think it's quite small actually, which is why I was sort of
You're already out.
can only see one of our dots. Oh, there we go. Yeah, yeah, he should be good.
Watching out.
Oh my gosh, he's about to get out.
Going out.
Ah.
Watching out.
Did you just hobble him on purpose?
We don't do things like that in Schmidt.
Very long 2 feet.
Not yesterday's fiasco.
He's a very speedy escape artist, this one. Okay, this guy's killing us. Yep. New plan. Too speedy for a quiver.
Watching out.
Now he goes to the
Okay. Right about speed. I mean, yeah. Let me see.
Thank you. Here's a bit of a trickster.
Make a move here.
Yeah.
All right, light speed then.
Roger.
Up 20 speed. I might just actually reducing the speed a little, kind of.
We'll catch up.
Imagine more than 3 times. Bridge, ROV. Is it possible to reduce speed 0.1, 0.2?
RV bridge, say again?
Are you able to reduce speed to 0.1, 0.2?
Sure, reducing speed.
I imagine we'll take a couple more samples here, so we'll try to piggy bank and prevent it from stop starting the whole way.
Alright, no
Cheers.
problem.
Thanks, man. Oh, late comer.
Yeah, thank you.
Chloe. There you go. So a little bit more time.
This looks like it could be a little bit of a hole. You could use a 50
Sure does.
Interesting.
Yeah.
I know everything that we have to do later, so that's weird. Like 6 years.
we have a special treat for you all this morning. We're going to have some rotating guest narrators, and these will be these will include all three of our honours students on board. So your first guest narrator will be Chloe Anderson, and she's an honours student at UWA and the Western Australian Museum. So I'm going to hand over to her first, and then I think after that there'll be Declan Stick, who's also an honours student at UWA and at AIMS, and then there'll be Amy Carmignani, who is an honours student at Curtin University, and then we'll finish up with Corey Wisson, a technical officer at the Western Australian Museum. So you're going to get to hear a diversity of voices this morning, and be nice to them, welcome them. All right, I'll hand over to Chloe now. See you guys.
Good morning, everyone. I know. I was in the big boots today. Calling the shots. Apparently I'm allowed to pick up whatever I want. So.
There you go. So you better give us some warnings. I better look on top.
It's about to be a long morning.
Oh, I thought you were being serious. That's good.
So yeah, he's cool, hey? But we've already got a sea star this morning. I'll show some restraint. Fishy. Should we do a sea cucumber flip? Look for some ulamids. Yes, this morning.
Roger.
Brilliant, RV. Can we come to a pause now, please?
Let me know stop.
Thank you.
I don't have quite the eye narrator does, but she always has a look at these sea cucumbers and looks for little tiny snails on them, the little host-specific parasites. So we're going to have a look, see if I can learn from the master, as it were.
It's all yours.
Thank you. How does this thing work?
You just turn this one on, the knob if you want to zoom in, zoom out.
Oh my goodness. Wrong way.
Slow turns. Slow is good.
Sadly, doesn't look like it. Alright. I think we're good, but thank you.
Go on.
Yeah, no, I think we're good. Didn't see anything on him. Thank you.
You're gonna put my head up. You don't want to. Sir, you're gonna put my head up.
No, I'll put it. So if you just keep going,
Yeah.
then I'll just let you pass.
Okay, yeah.
Bias the input that way and that way. What that wall looks like. We're just on 140, so that wall's probably 40 meters high.
Yeah.
Wow,
Is something?
that's cool. Oh no, that's just pretty.
Sponges.
Yeah, gorgeous. Oh, there's another sea cucumber. Should you have a look at him? Thank you. We got a little sea star there too. Got some cool stuff around here.
Against yours.
Thank you. Nope. This way? Yeah. That shimmer's back, hey? Always making my life difficult. Whoa.
Sorry.
You're okay. I was like, oh no, what did I do wrong? Oh, there's a shell. Doesn't look live. We can have a look. Ooh. Sorry guys, we're having some technical difficulties. All good? Thanks, guys. Now, where was I?
Is that a cylinder?
Hey Ellie, does that look like one of those solitary cup colours that we've been getting with a different colour? Yeah. Can that be worth grabbing because it's a different colour? I don't know. see some excitement for our 3-tone cucumber. That's very exciting. Glad to have you guys on board sharing our enthusiasm. Oh, it's so hard to tell. Yeah. It's hard to make calls on these things. Do you think we should pick him up?
Pick him up.
What do you think?
That's
Yeah. All right.
that's above my pay grade.
Well, I'm looking for little snails on him, and there's a lot of different like. Oh, awesome! Thanks, Narrator. All right, we're picking him up.
Yeah, I should fit.
You just want to pick them up and not sample them?
No, I think we'll sample him.
Okay. Well, which one is it? We've got a quick
Both of them. What did she say yesterday?
I'll fit it. It's small.
Yeah. All right. I think Narrator might want to see the underside of him. So we'll give that a go. Thanks, Barbara. You've obviously been watching along too and know exactly what Narrator would want us to do next. She would very much want us to have a little look at his underside.
Come a little closer. Oh, you got it. It's a cup coral.
Yeah, I was looking at that. It looks like a different colour to the ones that we've picked up already. Okay. She can hear me.
Do you want to see the bottom?
Yeah.
Shadow. There you go. Here's your photo.
Do you want to grab a photo of that, Nelly? Thank you.
You want to center it close.
Yeah.
Let's bring it pull up towards you.
Like this guy is probably worth sampling just because we can't really tell what's shell and what's possibly little eulemins in there. So it'd be good to have a little little scavenger hunt through him later and have a look. Hello. Yeah, looks like maybe these sort of ones are the right sort of shape, so I reckon it would be definitely worth. Yeah. Hi, Box. Thank you, Karen. Cool. We're gonna grab him. Let's do it.
Portugal.
I call the shots around here now. Students are taking over. That's okay. Just grab him while he's up there. Because we'll have a lot of face snaps of it. No dramas. Yes, 'tis the nature of science in Sectoid 3, as I'm sure you're aware. Oh, cool. Thanks, guys.
Good job.
Hi, Chris. Good morning.
I just have one.
Awesome. Thanks, guys. Good morning.
What are we doing? Just collect. I don't know. For a while. So I was drilling out the hole, the rest of it.
Oh, and happy Earth Day to those of you who it's still Earth Day for. It's rolled over to the Friday for us, but I'm sure there's still some of you that are clinging on to the last couple hours. Hope you've had a good one. Hope you've seen some of the really cool content that everyone's been putting out in celebration. Cool, thanks guys. Hi to my parents who are on the Facebook feed. Thank you, that was very exciting for me, getting my first sea cucumber, making my first call in the hot seat. Oh, awesome. Thanks to those of you who tuned into the Falkor's Earth Day live events. That's really cool. I'm glad to have you on board. Way. Got some pencil urchins coming up. They're pretty cool.
I guess actually this is probably sloping all the way up, so it should be a little bit of
Got a little friend down there saying hello. Oh, you're just a little lobster. Sorry, I should stop pointing if I don't want things. Another pencil urchin. They seem to be quite abundant down here.
that? Well, not just the title, on like the actual camera view.
Oh,
Generally, take it out. There
that's
you go.
much nicer.
Yeah.
Bridge, ROV, that's an updated waypoint.
Updated waypoint.
It's a nice little bit.
Don't tell me if you see anything too.
Co-captain.
Yeah, I forget when I got in. Yeah, what'd you do to your arm, buddy?
For like 2 weeks now.
I don't pay attention to you that much. Don't give yourself too much like value in my face. Yeah,
What's that ending of
sprain an elbow.
Yeah, me too. We're all terrible.
No, I was saying, it's been 2 weeks. It's the first time I've strapped it up because it's just not down.
Got a bit of a shimmer going on.
You know what that means? You need a roommate.
I've got a room.
Often when we do depth changes around here, we get this temperature change that creates this shimmer in the water, and it makes it very hard to focus on what's on the seafloor, but it's still pretty cool. There we go. Oh, that shell.
You want that shell?
Yes, please. Or at least look at it. Corey would kill me if we didn't stop for that. Do you remember what he's called?
one of those tiny ones.
Yeah.
Where is it?
I don't know. Nice.
Say sponge.
No, it's what's attached to the sponge that we're interested in. Am I right to touch things?
Go for it.
Anyway, it's this shell that I'm interested in. This guy.
Oh, okay.
Yeah. I know we've already collected one of these, but I think we're probably better safe than sorry since he's a live shell. I think we should grab him.
Or she, no sexist.
Everything's always dudes for me. It's always, we'll get him. It could be a she, right? Shelly the shell. You need an image of it? Sorry.
Stand by. I just might have to look a little closer for you, and I'm about to do that now so you get a better view.
You're right if we zoom back in quickly before you do stuff? No. That's okay.
I might get a little closer for you so you get a better picture of all this stuff.
Yeah? Awesome, thank you.
If I can.
I got this one.
Huh?
I got this one. Yeah.
Oh, beautiful. Cool. We'll be alright then. Hi Barbara. This looks like one of the spindle cowries that we've been quite interested in on this trip. Corey Wisson, one of the techs at the museum, is always very excited when we pull up shells, particularly if they're live ones. So I will be grabbing this so he doesn't hurt me later if he finds out I didn't. Oh, beautiful. Thanks, guys. Got that one, El? Did you get a sample in situ for that one? Yeah. Awesome, thank you. All right. You want him?
Yeah, you're not looking at your phone.
No, I was checking.
So it'd be great to get some really nice imagery and really sharp. So we need to set. I don't know how difficult it is.
We've got a couple of nice ones already. No.
We need to get some really.
Okay. There wasn't as much shimmer when we were doing it before. Ellie, can you get that really good photo that you took up with everything so? Looking at my phone, I spotted this. I nailed it.
It'd be interesting to collect the soft raw as well, so we have the food.
Okay, great.
I'm not sure there's many ones what it actually is. So
Yeah.
can we?
Yeah. Would we be able to grab the whole thing there?
So, sponge. I would like to just get a bit more imagery first, if that's okay. That's fine. Yeah. So we just want to sharpen it up a bit with the focus.
Oh, he's escaping.
And if you just hold, it'll go down a little bit. But when
Too much?
yeah, either go fully in, like get somewhere it's just the whole thing, but also zoom in now. It's in a lovely position.
Oh, wrong way.
Okay, that's right, we all do it.
Pulling a Karen.
Now let's try and focus in really sharp, but it's moving around a bit, so it's more tricky.
He's beautiful. Well, thanks, Ellie.
A little shrimp on it as well.
There's a little up there, yeah. That's nice.
Okay, cool.
And then get some zoomed out ones.
You want
Do you want to?
Hmm?
How fragile is it? Regular soft gel, just on
Can you take a photo of that as well? Thank you.
the shell itself is relatively good. It's not probably do this super soft touch, to be honest. It's yeah. I want to get this
Thanks for the support, Mum.
I can't tell until I grab a hold of it. It looks like
just we've had some of the animals have been in, but they physically fit because they can't look on the breakfast position. They're giraffe. So,
Giraffe. Yeah, that like patchy pattern.
I can scan it along neck.
Colour the giraffes out.
Just want to
It's a spindle carry. It's a giraffe. It's a giraffe.
almost put this on.
Well, it depends. He might need this other side.
Yeah. Cool. Thanks. Good work.
Thanks. Turns out I have made the right call and I will be expecting my promotion any day now. Just a good job. 16. Great.
Keep those snakes at bay.
Thanks for the support, Amy. You'll be in here later. You've already been announced if you didn't know. I think it's Declan and then you coming in. Tis the nature of science. It's always quite difficult trying to grab onto these quite delicate and quite small things, but we've got very talented pilots over here who always nail it. So I'm not worried in the slightest. Amazing. Beautiful. See the little shrimp on him there? He's got a friend. You can see his foot hanging out the bottom there. Yeah. Should be. Assuming it fits. It's okay. If you next to it click on the little comment thing. Yeah, it'll be fine. Yeah. Sorry, Ellie, just kidding. Ellie's going in a firebox. Just kidding. Beautiful. Oh, we got some brittle stars on there. It's a whole party. You just know that sea star's going to try and get out. It's terrible. Here's your chance. Go, go, go. Awesome, thanks guys.
What's that? Anyone plus 60. 60 that's a set.
We'll open up 20 as well.
Alright.
Cool, thanks guys.
We'll open 19, we'll swap it with let's go back.
What?
19, 20, and 21.
We've got insectoid rooting for the specimens. Interesting choice. Interesting stance to take. But yeah, he's been very fast. He almost got away from us before, so don't want to lose him again.
Which one you got?
19.
And Mum, I would have thought after watching 2 weeks of live streams you would have come to love sea cucumbers as much as we do, but clearly not. Okay. Get this show on the road, huh?
Yeah. Like whenever you retract it, that's like the word rams to do. Or we should be here in the back of us.
Good morning, Kat. And I'm absolutely with you, insectoid. There's always a part of me that hopes the animals win. But obviously, for my own selfish sake, I hope we win. That little fish. Good on you, Kat. For those of you just joining us like Kat is, oh, that's an interesting sea star. No, we got one like him, all good.
What?
All good, sorry. Yeah, for those of you just joining us like Kat, we are currently on our way between our 150 to our 100 metre transect. Slowly, because we keep getting distracted by cool things.
sure. I can press buttons. So it's all okay. Where's the button? Where's the logging gear? I'm not supposed to log things. There we go. Let's do. So that's all. Make sure.
Yeah. Sorry, this is one. I have to do the right.
2024. Wow.
I didn't actually read the year.
Exactly.
I just saw the month and the day and I got excited.
Another disco donut.
That's an updated waypoint.
Updated waypoint, thank you.
I've already got one of those today, so probably won't stop again, but they're always very cool.
Once you get zero to jump back 10, once you look at this chips, you see the 18 bar up. So it's a big part. It's not for Benthic projects. It's not for San Diego or something.
We didn't have 40.
They'll probably just get an 8 up now. So they make up new ones. Yeah, then Jason gets shit all up.
Okay, a bit of a shadow on the right side.
Yeah, further out.
And I was saying last night, I think this kind of brown would be good to try and do calibration. I don't know where Karen she's doing that thing. Fine, go over some.
What do you need for that?
But just an area where I have lots of potential type blocks, because I think this would be good. It's got this texture and unique kind of what you want. I'll go talk to Karen.
Yeah, no problem.
But I need to make a decision quite quickly.
Some octocorals. Octocoral. No, an octopus would have been very cool. Another pencil urchin.
I like how we took advantage of that.
He's all about friction. Every year's man breaks.
Yes, this is good timing for us to do it while they're doing that thing.
Okay,
So if we just hold there a second.
yeah.
I'll just go and work out. Sorry, Chloe.
Morning, Ian.
Morning, Chloe. So basically, if we imagine just imagine your little buttons there, or lots of tie points on the ground. So what I need to do is go over in every orientation and then cram them
Okay,
across.
we can do it.
Yeah, and just starting at the lowest altitude possible, and then we'll raise it a little bit. So whatever your lowest possible altitude, I'm going to check the
What we
photos.
can do,
So we need to do about a 10 metre strip.
Just make a grid.
we're going to make a grid, and then we're going to, we can't.
Here, I'll just do waypoints on his side on the lines. And you can still. What side did he want it? Side.
Oh, we'll do
We'll do something like this. We'll do a marker here, and then we'll do a marker over here.
yeah, but it doesn't drive to the markers.
No, but at least he can follow it.
Alright.
And then come up and then do another line
10,
back.
20, 30. He doesn't need that much. Just give him 30 meters.
So 10,
3 boxes.
30,
Then make a square.
and then 30 by 30.
Yeah, that'll be enough. Just doing a little calibration for the cameras.
Yeah, of course.
So it's just temporary.
No, this is great. I always love to see Ian at work.
Put a little, thanks, but we're going to be put it in there.
Yeah.
And then we'll just fly it.
Yeah,
Strips.
So Ian's another scientist with us on board the Falkor, and he does a lot of like seafloor mapping stuff, which is really cool. So normally when we go to a new location, he'll kind of start the day by going back and forth, kind of like you're mowing the lawn, and we'll be able to see kind of what all the features on the seafloor look like. It's often the way that we find super cool corals for Amy and Karen. It's my Ian's handiwork.
Thin strip.
Okay. So
So 10 by 3 meters.
yeah.
Yeah, try that. That's it. It's great. And if we just first try flying at the lowest possible altitude, then we'll go up and we'll do a few strips of that, and then we'll try. So I'm making notes of the start.
Get a show or something.
Actually, I can go to market.
Yeah, I wouldn't do that. It's, that's an unproven technology. It's just like my go home and then drone. I didn't.
For those of you watching yesterday, Ian managed to find us this beautiful bummy that had, it was about 4 meters tall, and it just had all of this beautiful biodiversity on it when we'd just been seeing basically sand up until then. And like some cool halometer and stuff, but it was just beautiful. So we're all very invested in where Ian would like us to go because we know it's going to be beautiful.
This one machine. There's one machine. Oh, you did? Oh, right. Okay, sorry. I'm still looking at the laptop.
Very fine. Did you get some nice images of this? Yeah. You know.
Exactly. We're just doing some Ian stuff. Oh, right.
It's good to get half a meter.
You won't actually get half a meter just because
It's much more.
Oh, yeah,
it's
no,
lower to the ground.
much limited.
Yeah,
If he does like a meter, you'll get like
so
Yeah, so like,
that's the camera's another meter and a half up. But, you know, so this might be a useful reference point as we go over it and the fish. Yeah, exactly, that's it, that's great, guys. So of course traditionally you kind of have a checkerboard, and I thought about what we did with checkerboards down there, but actually it can be logistically a little bit challenging also to have enough ground to get things over. So, but if you have a place where the software is very good at finding you know the tide points, but you just need to give it enough and enough cover. Oh, sorry. Maybe it's a bit. I should have stopped earlier when it's less of a slope.
and the whole thing is a slope.
And you need also slight changes in altitude to the seafloor, which is what you've got there with these things. Whatever that thing is above there.
He was online 6 minutes ago. I think he was. He was on Facebook 6 minutes ago.
Am I doing up and down, back and forth grid, or just up to the next marker and then like that?
yeah, come back here. You want to do it like this?
So
I can ask.
wait, up to the next marker and go, or
Fishy! That's cool to hear you've been doing a class on marine debris cap. That's awesome. I'm glad you're learning some new stuff. It's always good to keep up to date with the, you know, the issues that we're facing with the ocean at the moment. And you're very right. We are all squirrel people. We all get distracted.
Yep, you're kind of out of luck now.
Try the floods.
Yeah, it's flooding a few of the video though.
Maybe not his cameras though.
The floods were also
And the
I mean,
drain
cousins.
Try and move the arm further out, just give us a sec. Yeah, try that.
Yeah, and so almost go exactly over the top of the same track in the other direction. The width is not so a concern today, because it needs to see the same features, but this will be from a different orientation.
You try that shadow again. I've spun the arm right away.
Oh yeah, okay.
What a brilliant invention, Kat. The coral ball sounds amazing. We've got so many problems with microfibers coming through into our waters from our laundry. That's really awesome to hear that people are coming up with inventive solutions to deal with that. Are they expensive? Can't imagine. The best things are always on Kickstarter. Tell ya.
Yeah, if you can do a 180 and go back over again. So it's all going to sound rather particular and silly while we're going back and forth over such a featureless, well, to lots of other people, featureless.
Nah, it's cool.
But I could do with not many fish in the picture. I could do with some texture, some change in altitude on the seafloor. And it's, despite some of the video showing snow, actually down on the seafloor, we're actually not seeing much solid water column. So it's like an hour, 4 or 5, 6.
So why are we doing this again?
So this is this is an attempt at we're doing a few different ways to try and calibrate the cameras. So you can calibrate cameras above water, but what you really should do is calibrate them in situ to the kind of conditions that you're looking to extract the models from. And so,
Cool.
When you do it above ground, you have the luxury of knowing where you are. So,
Yeah, for sure.
but the key thing is just repeatedly going over the same area where there's some easily recognisable features. And so when you have a ground that has quite a bit of, I'm going to say knobbly bits, I can't think of anything more eloquent than other than that.
Some limestone substrate.
Yeah, some limestone substrate. I think this will work.
Structure.
Thank you. There we go. Where were you when I needed you? I can be as long or short as you need. I need at least another 10 minutes. Yeah. Okay, yeah. No problem. Yeah. All right.
Oh, okay. Yeah. Well, that's me, folks. Thank you so much for having me on today. It's been really nice. Thank you particularly to Kat, Insectoid, and my parents for keeping me entertained while we looked for stuff. We're going to hand over to Ian now. Lucky you guys, so you can hear a bit more about all of this seafloor mapping we're doing.
Thanks, Chloe. Yeah, sorry, my name is Dr. Ian Parnham. I'm with the Centre for Marine Science and Technology at Curtin University in Western Australia. So we have on board 2 still cameras mounted on SuBastian, and we've been playing around with some 3D reconstruction, working alongside my colleagues Andrew Woods at Curtin's Visualisation Centre, The Hive, and from our Spatial Sciences discipline at the surveying course in Curtin, David Belton and Petra Helmholtz, just trying to improve the ways that we reconstruct models underwater. This can be more of a challenge because of the conditions, and also normally not having reliable positioning, but in this instance actually we have pretty good positioning on SuBastian, which really helps. So what are we doing? We are going back and forth over what to some people would be a featureless seafloor, but for me it's one that doesn't have too many mobile objects and algae that's floating around in the around. That's going to get in the way of calibration. And so what we're looking for is for lots of different unique points on the seafloor that we'll be able to match the different photos as we go back and forth. And from that we were able to then maybe estimate and calculate the different camera coefficients that need to be estimated, particularly things like correction for radial distortion. So because you don't know until it comes up, we might be quite particular, taking more, better to have redundancy and taking more imagery than we need. And so that's what we're going to do. So if we do one more pass back over there, if that's okay, and then we're going to do one crabbing after that. So we're going to first, so the 2 kind of different variables I'm looking at doing it is to have it in different directions and orientations. So we've been going back and forth at 180. We're then going to turn 90 degrees and after this transect and crab across. And the other one would be to try and do this again just at a slightly different altitude of the vehicle. I'm going to go in. Is the current a problem at the moment, or just? I'm sorry. So here we go for another pass. And that would have been, after this that would have been 2 in each direction, at least.
Yes.
Well, yeah. As I'm going to be also going to check out the quality of the pictures coming through. I've also got with me Declan, who might come and introduce himself, and if you don't mind, Declan, take over a bit on the mic. So I'll just so you might want to completely introduce yourself.
Yeah.
Thanks, Declan.
Hi everyone, I'm Declan. I'll be taking over the dive to give the other scientists a little break for the next while. I'm an honours student at the University of Western Australia and the Australian Institute of Marine Science. My primary research this year is on the spatial variance of coral microbiomes at the Rowley Shoals. But on this dive, I've been helping out with eDNA collection, which has been used with the video transects to survey some of the biodiversity here at the Ashmore Reef. I'm also involved in some of the population genetic studies we've been doing, so helping Chief Scientist Karen Miller. So we've been looking at some of the Leptocerus and some of the other species of hard coral to try and understand how they're all linked. For those who are just tuning in, this is our 13th dive. We've been conducting video transects today. We're doing a 150 meter transect, and we're making our way currently to our 100 meter transect, where we'll be doing another video transect and possibly afterwards collecting some biological samples. So keep your eyes peeled for anything interesting. Make sure you comment so we can video in and scan for anything that might look cool. We can try and collect that if it's worth surveying. We also have the wonderful artist on board, Ellie, who's on the sea log, who'll be taking pictures and recording the samples that we collect. So thanks to Ellie.
Nice to be photos. Imagine how we're taking the photo and do an orientation. That'd be great.
So just there and back.
Yeah, there and back, and we might flip it. So,
it's wonderful seeing the transition from substrate today. At 150 meters, it was kind of desolate sand, and as we made our way towards the 100 meter mark, we've seen the substrate become a lot more rocky, a lot more diverse, a lot more animals and critters crawling around. So it's cool that we're getting a lot more stuff to look at now. I'll tell you what, at 6 a.m. this morning, it was looking very desolate. Had a huge krill ball, which was interesting. Hopefully that didn't contaminate too much of the eDNA analysis, but I'm sure I'll be filtering out a lot of krill from my water samples later. Thank you, sir. Enjoy your tea. Thank you.
This direction seems to have kicked up.
Well, there's quite a bit. There's quite a bit of
It's funny. It was okay. Was it better further deeper? Was that, or is it just the tide has changed?
It's really hard. So
that's something I should have done earlier.
But we're back.
Oh, we collected that Benthic 10 before.
What time's time today?
Awesome. Did you collect the 10 4? Oh, the old man collected the 10 4?
How we do that.
Oh, okay.
I did the
So we're thinking actually once we get to high tide, maybe we'll slack off a little bit before it goes out again. Okay, so that's else. So it might not worth doing. Yeah, that's approved. I don't know whatever you did. Is it the current that's good. So I assume one direction is a little bit quicker than the other, is it?
It's just where the boat is, where the current is, you just get pulled around. Right now, it's fun. No. That's what's happening right there.
Ah, okay, so it's just too hard to do at the moment. We'll do it later.
Sure.
I don't know.
Yeah, I'm just gonna.
Can we, as we're here, can we just drive through the middle of it from this
It's
direction?
just probably best if we just take control here for a
Hold on.
Yeah, okay. Sorry, yeah.
Are you doing it?
Yeah, we're just trying to do calibration,
Yeah.
just to intensive survey area.
Yeah.
The current sort of colour of this.
No. I got it.
Something pretending to be a sensor.
Dicks.
Just tell me what you want.
I'm fine. Just let it go. This direction, what you can do it is easier. It's even better to it.
That's going well.
Pretty far away.
Could be on this one. We can go whatever orientation you like, but just ever so slightly higher.
Yeah, no problem.
It's just go directly over
Yeah.
the production lines. said hello to you. You see on the message?
Yes, I did see. Some red guy says, "Hi, Declan." Well, hello to you. Thanks for joining us this morning. Hopefully we'll be making our way to our video transect later and we'll be able to see some cool critters.
90 degree, or just crab again. And then we'll try the 90 degree, whatever's easiest.
As I've done.
Okay, so if we try at 90 degrees at that altitude, back that way, and then once the other way, and then we're done. Do two more lines at this altitude, put it there.
So we're just having a data log switch, so thank you to Ellie for your work this morning. Amy's now joining us for the next little stint of the journey. And shout out to Corey as well, who's back there from WA Museums, looking out for the gastropods and mollusks of the trip. He's done a very good job so far.
How's the dive been so far?
I've only been here for the last 10 minutes, but this morning I was here from 6 AM and didn't see much. Saw a shark, which is pretty cool. Lots of krill balls.
Oh yeah.
Then we picked up a Benthic tenaphore. Got another spindle carry, had a few of those earlier on
I
the
saw
trip.
the spindle carry.
And another sea cucumber for the collection.
Yes, I think that small gastropods.
That had some small gastropods on it, so that's why we've collected that one.
Okay, we just need to. We'll just do one more and head up. Could we do it through the middle of those? You see, there's that little gap between those bunch and the one up there. If we just do it in between those, and then I'll actually head up.
The one on the top was just me going by, but
Sorry, is that a question?
I don't know if the one on the top was actually like part of the actual survey, but I can fill it in if you like.
Okay. Yeah, I see what you mean. Yeah.
These ones go in certain houses. These guys here are just switched to like this way, just like that.
Good morning.
See one above north of this. Just a little bit.
Yeah, just there. That's fine. It's pushing you, isn't it?
Yep.
Yeah, can do a zigzag, that's fine.
I'm gonna go back a little bit.
Yeah, should we try, do you mind if we do go there and back again? Yeah, that's fine. Sorry, I'm asking a lot in this code. I can see how those answers. Might as well get.
How's the terrain looking at this site in?
It's kind of useful for what I want, so that's why I've kind of picked it. When you get kind of a lot of stuff growing on it that moves around, there are bits, but there's not a lot. It'll make it easier. It might not be the prettiest mosaic to look at the end, but it might be the most useful to calibrate the cameras on board, and then you apply those calibrations to the other images.
Cool. So have we sort of mapped the 50 to 70 meter area? Does it look like there's some rocky substrate down there where there could be some coral growing?
I haven't looked at that. That was last night. But what we're about to come up to is the 100 meter, where we're on effectively kind of an edge. I think you can see on the map there, we're going to be along that edge, and I'm hopeful that probably is a regime with there's a lot of current streaming past it that filter feeders might enjoy living on there, and so we'll find out.
My bet is on some big Gorgonian fans.
Big Gorgonian fans.
Sponges.
We might see a little pygmy seahorse today, fingers crossed. A little second last dive treat.
That's it. So if we're on an edge, do you know how high that edge is to get to the top of it?
Well, in the transect's at 100 meters, and we are currently sitting at 132, so we're going to have to go up 30 meters. 32 meters. So that would be
So are we on the edge now?
No, we're about the bottom. Can you see on the map?
Yeah.
So that's where it'll look on the right hand
Do
side.
you know how high the edge goes though? I'm just thinking.
Oh, we're going to be on the top.
If we were to pop up onto
Yeah, we're going to be right on top.
Oh, okay. Oh, fantastic. That
Yeah.
sounds a bit corally to me.
Okay. Might be why we chose it.
Possibly. Possibly, yeah.
Possibly. Who knows? Thank you very much, guys. I've probably got. I think that's enough time there. Whenever unless there's a bit that you think is there anything that you think we'd worth?
I was gonna go the middle.
Yeah, if you don't. Yeah, if you think you can.
so
I just feel bad.
how
I
do you
see
want
these
it?
kind of currents.
How high do you want it?
So we're struggling to actually
Yeah, that
see
little gap.
any of the
How high do you
floor.
want it?
Not that there was much
Whatever's the
at
most comfortable to
150
drive
inches.
at. I mean, ideally close would be
Desolate,
good.
okay.
Just going to see how the sort of
So we'll be shortly finishing our calibrations and heading to the 100-meter mark for our video transects. And then on the way, we're going to be looking out for any biological samples that we might want to collect. So keep your eyes peeled. Let us know in the comments if you see anything interesting or fun. Oh, a new rock.
It does not want to behave, doesn't it? How would you describe driving an ROV in this kind of current? Is it, I don't know, is it like driving a car on oil slicks or something, you know, like, or on ice with no grip, while someone's trying to push it in the other direction?
It depends basically like ship positioning versus currents and all that, and which direction we're heading. So like whenever we were with our back to the current, trying to keep that from pushing us forward is a lot harder than when you're pushing into it.
Yeah, I can imagine speaking some cars there. Yeah, we're now just to fill in our gaps. And we can go zigzag if that's easier. It's easier to zigzag through. It's not a problem. Thank you so much, guys, for your assistance. I just sometimes get odd requests to just mow up and down the same bit of featureless seafloor might seem a strange occupation.
It might be a loading issue.
Zigzag down.
It's easier to take land photos. She has plus a photo of the specimen and the lake, so I could see. Yeah, just for sure. We have a lot of stuff that comes off of hosts.
Oh, that's satisfying.
So I do that host soft coral, and I copy those into I can copy it all the way to like what the came off the yeah, for all that stuff. Yes, and it's come off those and so on. So just takes
Well, this will keep me busy for the rest of the day. Looking at this. I'll just do the zigzagging through it if that's easier than trying to
you're
pick
saying
line.
zigzag or do you want a diagonal?
I'll just go where there's holes. Just imagine you're playing a computer game and just try and fill in some of those holes, but slowly, gently through.
very little robin fish. Once they glide along the bottom of the surface, they look like they're having wings. Very cool.
Nice done. So Declan will be done almost in a second, then we're heading up
Fantastic.
to the 100 metre side.
Yep.
What are you hoping to see up at the next the 100 metres? Is there anything particular you think?
Well, as with all sites, I'm always interested in coral, so any coral we can find is always good. Especially the Leptoceras for the population genetic studies would be fantastic, but we're hoping to see anything really. Anything hard and corally would be great for any of our biological surveys. We're also hoping to find possibly some more benthic ctenophores. They haven't been sampled much at these depths, so it's fantastic to be able to see those and sample some of those on our trip so far.
All right. Thank you very much, guys.
Thank you, Ian.
No worries, Declan. So you can see where we are going. How long do you think it would take to transit up there? It should be interesting going up the slope, so keep your eye out for things.
Yep, make sure we comment if we see anything interesting or to help me spot it.
Thank you very much again, guys. You did a great job.
Brilliant, Jeremy.
Great job.
Bridge.
Hey, we can continue to that waypoint.
Okay, continuing to waypoint.
And then I'll update you probably one more time.
Alright, thanks.
Alright, here we go. It's a sea star. Lots of sea stars.
That's when they start building.
I think that's what's that?
Messing everything up.
When we get to the waypoint and put the section sign, let's drag it.
Does that look like a new sea star?
It's fine coming up here.
What do you want to look at? Declan?
I want to get a video.
Yeah, we'll have a little zoom up on it. That's
what,
all right.
What do you want to look at?
Yeah, just the sea star on the left. I think we've probably already sampled it, but just in case, we'll have a little look-see.
Go for it. No, just step after that. You can look at it.
Yep. So down is up.
Down is up, and up is down, or away from me.
And then to zoom in.
To the right, John. Spin it.
Oh, you spin it?
Yep.
Take your POI.
Yeah, take a POI. That would be great. Does it look like we've sampled something for it?
I think so.
Seems relatively familiar. So we're just going to mark a waypoint and we might come back to this one.
A waypoint here?
Yeah, that'd be fantastic.
Not sure
does look vaguely familiar. Cool. Alright, got that.
Okay, guys, we're almost ready to roll in here.
Alright, we can keep going.
Carry on.
Yeah, thank you. Thanks, huh? Can you guys work it out? All jokes. Fantastic. Thank you very much.
Hey, Jay Rod, if you guys want to answer the question. What?
I don't hear anything.
That's awesome. Are there any more questions, or shall we go to Chloe?
There's quite a few sea stars here. That is a huge urchin.
Definitely shortcut that one.
That's huge.
I don't hear that.
This is interesting.
Structure coming up.
Yeah, it's a nice structure here.
John's answer.
What do you reckon? Pop down and have a look, Corey?
Yeah, I think
Yeah, let's pop down and have a quick little look at this structure. See if we can find anything interesting. Looks like there's a little solitary coral at the base there already.
No?
Alright, go ahead.
Yep.
Go up here and then just go to the computer. Guys, what was that? How was
Yeah,
the
it's a nice
robot
little
built?
solitary
how
coral.
was the robot built? Maybe it's a good question. We have a whole YouTube series on how the robot was built. The robot is a custom science ROV, and so a team of engineers came together.
I think we're done with the solitary corals, but always nice to have a look at. Yeah, we'll try and have a look a little. Keep your eyes peeled for any nudie breaks. There is a nice starfish, which I can see up. Yeah, sea star.
With that, are we ready to go to the tour and take a bit of a tour? We'll do time for questions a little bit as well.
I don't recognise the sea star.
Hi guys, how you going?
Take a frame grab of that one.
Well, my name's Chloe. I am an honours student at the University of Western Australia, and I'm completing an honours project at the Western Australian Museum. I'm here as a research assistant on the RV Falkor. This is where our students spend most of our time. So this is the control room. This is Captain Declan doing his thing. So yeah, we spend most of our days in here on our dives. This is all the live footage that we're getting straight from the ROV SuBastian down on Ashmore Reef. We're seeing a very cool sea star at the moment that hopefully we're grabbing because I love sea stars. Yeah, so we spend most of our day down here. We spend upwards of 12 hours down on the seafloor looking for cool little critters that we can pick up, anything that's interesting for us to study. We've got our pilots over here. They do all of the, it's basically like a remote control car. So they'll sit with a little joystick and drive their way around for us, and then we'll yell at them if we'd like. to have a better look at something. We've got some really cool stuff going on the screen at the moment, I hope you can see that. And then over here is where we do sea log. So this is where we create a database of all of the stuff that we're seeing. So we're able to take note of the depth of times, of what we've seen, what we've collected, what we've left behind. You can see Corey beautifully pointing out some subcorals and sponges and things as we go past. Go, the boys at work. This is Jerry Rod that's working out here at the moment. Jerry Rod. James over here working hard as well.
So Chloe, can you clarify for everyone, is it a special job driving the ROV?
It is one of the most special jobs I've ever seen, to be honest. Everyone's nodding his head away. Yeah, they have such a cool job. It's not an easy job. It's actually quite hard to pick up some of these really small and delicate things, but they always seem to find a way. They're very talented, all of them. But yeah, they're doing some really, really important work for us. Without them, we wouldn't be able to do the science that we get to do.
Chloe, what does a typical shift in a control room look like for you? How long are you there and what are you doing there?
Good question. So we do most of the time a 2-on, 2-off roster, so we'll be in here for 2 hours at a time. And that's when we're doing sea log. We're coming to the end of the trip now, so we're kind of getting promoted a little bit. Some of us have had a chance to get onto the captain's chair, like Declan is at the moment. Hi Declan, camera's over here. But yeah, most of us are sitting on sea log, gathering all of the data for us to look at later, particularly if we're picking up things that we're intending to have a look at later. We've got to make sure that we know exactly where we found it, what we found it looking like, because it can often look different once we've got it back to the lab and preserved it.
Do you feel like you're underwater when you're in the control room? Would it like make you feel like you're down there?
A little bit. It's really dark and warm and cozy in here. When you're staring at an ocean screen all day, it does feel quite comfortable. It does feel like you always want to scoop it up like you're floating around. But there is that little added extra of we have to work, we have to be training our eyes to look at things all the time, so it's not just relaxing.
Does anyone have questions about the control room? Before we move on to
You know what? We really should. We've got a couple of meters of like the Nautilusometer, and we'll put up how many we've seen. But there's definitely a sense of pride when you find something that somebody else hasn't. Declan's very good at it. We don't always have a 100% hit rate, but we do our best to spot the cool things. It's often a nudie hunt with us. We're looking for little nudibranchs, the little sea slugs. Because Narrator Wilson, who you met before, loves them very much, and we always get brownie points if we find one.
was that J-Rod just waving the robot arm at him?
Sure is. Does anyone have any questions for J-Rod? We can probably chuck him one.
How big is the robot?
How big is the robot?
How big is the robot?
It's about the size of a small car.
Dara says about the size of a small car. We call it a little bus most of the time because it's so square shaped. We will actually go out and have a look at it at the moment if you'd like. So,
It's down here.
We go to the aft deck?
yeah, go see where
We're not gonna see if I can.
I think we have one more question for the
Lancers we got flying.
Yeah, let's do that.
Yeah, we'll go see where it's coming from.
They would like to know if do you see any crocodiles?
Crocodiles? Not yet. Not yet. I kind of personally been
Red.
Alright, we just had Chloe running a little ship to shore, which is why we had a bit of background noise then. Talking to some school kids about what we're doing here on the RV Falkor. So we're just going to continue our search and we're going to make our way to the 100-meter transect still. seeing a lot more sponge and diversity as we make our way closer to the 100-meter mark.
Yeah, that's like July 7th. Yeah. So hopefully I'll take ownership between Hawaii and San Diego. We'll do a road trip to San Diego.
We're at the base.
Cool. This is a nice spot.
It's time to start climbing.
You want to look at anything before we go up?
Base camp.
Do you want to look at any of these closely before we go up?
Yeah, let's hop down somewhere and have a quick squiz. Looks quite nice around here. Anyway, that's nice to land. Just consume it anyway.
I don't know. I just figured it was an Australian thing.
What was the word that you used?
Squiz.
Squiz?
Have a little squiz. Haven't heard that before?
No.
It's like have a little look. Have a little squiz.
Aussie slang.
Yeah. Sorry, Australian slang for all our other viewers.
I heard some new ones last night as well.
Thank you.
Fair dinkum.
Fair dinkum.
Struth.
Struth.
Didn't know it's truth.
Narrator made out like this was very hard to control. I don't think it's too bad.
You're a natural deck.
What does the front look like? Front of the boat, what does it look like?
It's a nice crinoid.
How come Sebastian doesn't break up the water?
Just bring the water.
I think that's just a sponge. It's a purple wormy thing.
What is that? Oh, center screen.
Yeah.
Zoom in a little bit.
That's max zoom, unfortunately.
Maximum effort.
I can screw up
I wonder what that is.
Just a quick question, can everyone, the picture of Sebastian or the picture of Falkor right now?
How does Sebastian move?
Chop chops.
Wetland wall control room. Wetland boat.
How long did it take to build
So you reckon,
Sebastian?
should we check out that purple?
That road.
Not quite sure what it is.
You have to move the arrow moving closer.
Okay, you don't need more.
Probably a sponge.
Oh, here's one.
See what you're looking at?
It wasn't really moving.
No, I don't think it was moving. Nah, I don't think it's anything. We'll look over to the right.
Took things apart, put it back together.
It's an interesting slash coral.
That's amazing. Like, where does one start and the other end?
I don't know. Like that's sponges just growing sort of over the base of the coral.
Yeah. Very cool.
Looks a bit like a fancy chair.
A shower head.
Yeah. Oh, that's good actually.
Yeah. Oh my god. So what we found?
Oh, so this
Nice
is a
soft coral.
soft optocoralia.
Very nice soft coral.
You need that quite a bit, yeah.
Yes.
Yeah, nice.
How far does Sebastian go out to sea?
I mean, probably not old enough to need a shower chair.
Like how far would
That's
you
on the down.
Yeah.
You know what I mean? It's like, yeah, I don't need one, but if there's one in there,
Yeah.
why not?
Wouldn't say no.
Now, have you got your caption shots? Are you waiting to take it?
200 meters.
I want to get this stuff.
Use that as a call, right?
200 meters would be daring.
I've done more. I've done 350.
I've never been more than 250.
We had to.
Okay, so
What kind of animals have you found? Sorry, not again. What kind of animals do you find?
Actually, a great segue for where we are right now.
Lots of stream to show.
Where are you right now?
Okay, I'm happy.
okay.
Yep. Onwards. Thanks, guys.
Well, sorry, when does Sebastian contract?
Whatever we're allowed to.
What's the biggest animal that you found?
We found a whale shark the other day.
What's the biggest animal you found, J-Rod?
Mass wise or length?
Let's go with mass.
Probably the waterfall that we went to in, off the coast of San Diego, Rosebud.
Cool.
Not live animal, of course.
Yeah.
It was still
Impressive. Yeah.
Smelled really bad. And it smelled really bad.
Western Australia, and they saw a 50-meter long photogrammetry, which is
150.
like a jellyfish,
Right?
but
You were there.
Yeah, I measured it.
and it's estimated to be the longest sea creature ever seen.
150 meters.
Did you find anything strange?
Have you found anything strange?
have you been a marine biologist for?
Getting a lot more activity now, lots more sponges as we're making our way towards the 100-meter mark. Hopefully we can start seeing some corals, which would be great for our coral team here. Amy and Cameron would be delighted if we found some aquatora.
I would. So roughly how far are we from the transect, guys?
30
Roughly?
meters.
Time-wise?
I can get there in 2
45
minutes.
minutes. 45 meters.
Oh, 45 meters.
Yeah, but
45 minutes.
Not too far.
if we keep stopping,
I can get there in like just a couple minutes if you want to start that. Amy.
We'll just take
Leisurely stroll our way there.
What's the #1 dive rule? Don't go fast underwater. You'll miss things.
Something on the left there, looking kind of snaky-like. Could we take a look at that?
What's that?
Just back there on the left, it looked like it was something interesting, but possibly snake-like. Could just be a stick. Looks like it
on
might
the
be a
left?
stick. Yeah.
Declan Stick finds a stick.
Declan Stick finds a stick. I'm very biased with my scientific collection. Yeah. Don't worry, guys. It's just a stick.
Could be a
Keep
very
on.
important stick.
Could be a very important stick. Just a stick.
what did you say? 130 meters, right? Is that,
Yeah.
what is that?
410.
26.
426.
Ooh, that looks pretty.
Have you seen anything being eaten? This must be a thing to eat.
As a kid.
What's the habitat?
fish coming up.
It's a sponge in that.
What's that?
No, it's not. I think it's just a sponge on the right. Just tossing it between. It's always, is it a coral or is it a sponge? 90% of the time, it's a sponge.
It's like almost identical to the shape of and colour of the Leptoceras.
It is.
Very hard. Ooh, it's nice.
Yeah.
Something really about it.
Yeah, it looks like a sponge. It's almost metallic. It's really cool.
Yeah, that's really cool. Interesting how the fish, they sort of only hang out in the cave bit.
Don't go to those.
Doing great with the maps there,
Yeah, I'm very good at zooming. It's just looking promising.
Alright, take a look around out here. There's a bunch of stuff.
Yeah, I'd love to have a little
go over to the transect.
little squiz around here.
Yep.
Get up to some of those sea fans.
Yeah, we're definitely gonna have a look around.
On some speed of parrots.
What you got one this morning?
We did it.
Yeah, we got a spindle this morning. Right, let's keep our eyes peeled for anything interesting.
Take a closer look at all these fans real fast before we
Yeah.
fly over. Alright, let's sit down right in front.
Thank you. Looks like there's a nice big log in the background as well. Beautiful.
It's all yours then.
Thank you.
There's all that we can look at whenever we
Yeah.
as well. So.
There's lots of stuff here. It's really cool. Corey's just joined us in the co-pilot seat. How are you, Corey?
I'm good this morning, Declan. How are you?
Fantastic. Finding a lot more things here as we're making our way towards the 100 metre mark. It's getting a lot more interesting.
It's definitely getting a lot more busy. Hopefully we can spot a spindle carrier here before starting the 100 metre transect.
Yeah.
Making our way there this morning.
Yeah, big fan of the spindle carries. Absolutely magnificent shells.
We found some really interesting ones this trip. So hopefully when we get back to the WA Museum we can look at them more closely and hopefully put some through for DNA sequencing and start to paint a picture about the species we've got here at Ashmore Reef. And there's certainly every chance at least a couple of them may be new, we're hoping. So more research needed, but certainly the ROV is allowing us to pick up these animals with relative ease, opposed to spotting them diving, you know, at shallow depth where you've got a bit of limited time and then not as great visibility as the cameras that you're seeing today. So beautiful, beautiful gastropods they are.
Have a look at this one as well. So what are you hoping to find today, Corey?
Well, considering this is our last site for the trip, I think I've got that right. We've got
Yes,
today's
this is site 7.
Yep, and tomorrow will be our final dive. So it's a bit of a big ask, but we're really hoping to pick up the Baylor shells we've mentioned many times.
Is that the Milo?
The Milo, yeah. So only known by shell from Ashmore. It's endemic to Ashmore. We really want a live specimen for a number of reasons, but mostly so we can test whether it is in fact a separate species from the mainland ones that we know. There's another species called Milo amphora, which is the northern species, and then as you get south around Perth, the southern Baylor, or Milo siltonus, is in that particular area. So there are a couple of other northern species as you make your way around through the Indo-West Pacific across to Queensland, but this Ashmore species was recently described, relatively recently, I think 2005 or thereabouts, based on shells, and it certainly looks different. There's some differences, but the genetic work will really help clarify whether it is in fact different. I think it's probably most closely related to Milo amphora, which you might expect, which is the one on the adjacent mainland in the Kimberley region. But that's yet to be, as I mentioned, fully distinguished, and hopefully we can pick up a live crawling Baylor shell, and of course we'll get to see the foot as well and get an idea of the pattern and the colours, which is another character which can be used in some groups.
Well, fingers crossed. Hopefully we can find Milo lurking about. Today's transect would be a fantastic grab on our second last day here.
It would be fantastic. It would be certainly a huge highlight. So Ashmore Reef is quite a remote place, as we've mentioned. I'm not sure how many people have looked up the reef on a map, but for us it was 2 full days steaming in RV Falkor from Darwin, which is in the adjacent state called the Northern Territory, and we departed there, and it was 2 full days steaming to arrive at Ashmore Reef. So that gives you an idea of just how far this reef is from Australia and
Can we do a gentle orbit if we're just looking? We're not sampling. Are we sampling anything just here?
No, we're just looking around for stuff. But if you
Can we
need
do
to
a,
do
can we
an
do a
orbit,
gentle orbit around this sponge, using that as a centre target? Is that
For a
possible?
3D reconstruction.
Maybe, yeah.
Maybe,
Who
yeah,
knows? If you're just, and also give you a chance to have a little look around. The fans are quite a struggle to reconstruct. The sponges work quite well, but you need something from all of the little angles. So just have to be gentle. Orbits around.
Yep, so we're just going to circle around the sponge so Ian can do a 3D model for his work. We'll keep our eyes peeled for anything interesting while we're going around.
And the other things I guess I'm hoping to get in the last day and a half is some live carries would be great. Certainly a favourite of mine and a lot of shell people. Carries are always quite popular.
Smooth, beautiful.
Lovely, yes. They're what keeps them so shiny is their mantle comes out and comes up over the. So the mantle is a special, specialised tissue that all mollusks have that effectively creates the shell, lays down the shell. So in the case of the carry, it will come up over the shell and in many ways acts as a car wash, car polish, and it keeps it nice and shiny. So that's why you see these strikingly beautiful shiny shells, along with the spindle carry, which is closely related, has a similar process with the mantle coming over the shell.
The other day, our artist Ellie did a similar thing to what we are doing at the moment. We're circling around a sponge to create an image 3D model, which you can use for some of her mugs, which she might be creating later on. So reconstructing these beautiful sponge molds, so she can create some beautiful art with it. The branchy thing.
Yeah. What's that?
This?
So the non-biologist.
A log.
Yes. What's that?
That's a log.
It
I
literally
think
is.
we call it a logus.
Logus.
Spadosia.
Oh, it's a bit of natural.
I don't know, I think it might be a bit of a sponge actually.
Maybe.
Yeah. It looks sort of a sponge-like.
Pressing on to
the transect?
Yeah.
Yep.
Hold on, I'll spin around the other way real fast.
Thank
I
you,
gotta
guys.
take a turn out anyway, so I'll spin back.
When you're ready, Chris, we'll...
Hold on, I gotta take a turn out, so I'm gonna just spin around this again the other way.
No worries.
Stunning seeing a lot more diversity now compared to where we started this morning, which was these desolate sort of sand dunes. And as we've made our way up the bank, up towards the 100-metre depth, we've got a lot more sponges, fans, a lot more diversity, biodiversity throughout these areas. It's a lot more interesting to look at now.
It's like a rabbit head. It's the angle on that sponge. it turns out. Go.
Now a game of sponge or coral.
Perfect little donut you did there.
They turn out fairly symmetrical, all things considered. Looking at it from there.
That's a nice looking sponge on the right.
Yeah, it's beautiful.
You go closer, you want this angle going. Sorry.
Oh wait, the transect start?
You want this?
Cool.
You want closer? You
Start
want,
transect, guys, please.
do you want any closer or is this fine?
Starting transect.
A little bit close would be great if we can. That's fine, thanks. Beautiful. Alright. Thank you to Amy and Chloe, our video loggers. Doing a great job on the sea log, making sure we're getting nice photos and beautiful data to observe later.
So we're probably about on the pretty well eastern
Yes,
side of
east side.
Ashmore. Yeah. Question, where are the fish? Hopefully, as we make our way along the transect, the instruction we might start to see
Starting the transect.
more populations
Alright,
of fish.
you just
starting
want us to
appear.
go south continuously at 0.3 knots or something like that?
Yes, that'll work. Yeah, quite very well.
Okay.
Certainly been a lot of interest with the visual records of the fish we're seeing, along with sea snakes and other individuals. So a lot of interested people following on YouTube and various institutions, and really liking access to the live stream as a way of seeing what's living around the reef. It's interesting. Hope you're enjoying the footage today.
If there's anything you want to come back to, just let us know. We'll come
Yeah,
back
of course. Thanks.
very lucky with weather. I believe this time is relatively good weather, but even still,
It's been stunning.
we've had some fantastic weather. Swell's been relatively low. It's made our work really comfortable. And, we're hoping that stays the same until we make our way back to Darwin, although I believe the wind it may be picking up for the journey home.
Considering we avoided 3 cyclones at the start of the trip, I think we've been very lucky.
Are we doing Niskins halfway?
Yes, we will be starting the Niskins.
We're gonna do all 6 today or just 3?
We've done 3 at the 150, so
Okay.
just another 3 here.
Great job today.
So hard to tell these are.
Yeah. What are we at? So yeah, about 100, 103 metres. Yeah.
Is it a cucumber?
It does look like
Not convinced.
yeah, right. Just
It looks like the base of the fan.
yeah. So maybe Declan, you can talk through what happens for the DNA projects once we leave Darwin, I guess, and how everything gets transported back and so forth.
Yeah. Unfortunately, the EDNA is not my specialty area, but I've been lucky enough to do a lot of the sampling on the boat. So halfway along this transect, we release 3 of our Niskins, which will fill up with water, and then I'm taking 1 litre from each of those. So the idea is to use the water samples in conjunction with the video transects to get an idea of the biodiversity in these areas. So because our video transects are done at those 3 different depths, we can sort of get an idea of the diversity of each of those areas and compare it between. But eDNA is sort of still quite a new field. Lots of the techniques are still being refined, so they need to be worked with other methods, other survey methods like video transects to make sure the quality's there. So once the samples are up on boat and the submarine brings them up, I'm just filtering them through a millipore, I think it's a 0.5 nanometer sort of filter membrane, which basically stops the DNA on the membrane so that can be rolled up. And so that's kept at a very cool -80 until it's transported back to the labs. Not exactly sure of the methods. I'm pretty sure we'll be doing some sort of PCR followed by a MiSeq Illumina sequencing, and that'll give us an idea of the abundance of DNA in these areas, and that can be related to the diversity later on.
Fantastic.
Yeah.
So is this something that's becoming more common in various areas for sort of surveying biodiversity?
For water, mainly at the moment. I know eDNA is sort of being used. You can use it to collect scat, and so there's been studies with migratory birds because they are obviously quite hard to study. So taking their fresh scat samples can be a way of monitoring what they're eating.
Yes.
So that's one way that eDNA is being refined at the moment. But yeah, lots of the work at the moment is sort of around water bodies, mainly streams, because they're flowing, and you can instead of just grabbing a liter of water, you can put down the filter membranes. You can attach it to almost like a grid system and have a few different membranes. If you put that into the river, the water will flow through the membranes and pick up all that environmental DNA.
Yes.
So you just leave it in there to soak for a few hours and then take it out and you can sort of get an idea of the diversity. But again, because there's still a few issues around eDNA and DNA degradation. Obviously, water bodies are constantly shifting, so they might not reflect the diversity of what's actually in that environment. You also get a lot of dead organisms still shedding cells and DNA into the system, so you have to be really careful about saying, relating that abundance of DNA to the actual diversity present in a certain area. Which is why I've been quite careful in some of our comments. You see we come down and do our eDNA samples at 150 metres. There's a lot of darkness still, and so when we have our lights on, there'll be lots of krill and microorganisms flying around the sub. So if we release our Niskins then, we're going to get a lot of bias towards the DNA present in the sample.
Yes.
So we've been turning off the lights for a little bit, letting the krill sort of disperse, and then taking our water samples, just so we don't get that huge abundance of krill DNA in the water sample. I also might run a second filter, just to filter it another time in those areas of heavily abundant krill. Yeah, so it's a really exciting survey method. Certainly has a lot of refining to go, but it's very promising, and I think it's going to be a really good way to survey biodiversity once we have those techniques down in the future, especially for marine-based environments where it's very hard to pick up cryptic species and lots of small microorganisms that are very hard to survey normally. So it's a very promising technique in our field. Very exciting time to be doing it. Very happy to be in these initial sort of stages of sampling. It's great, especially as a uni student currently learning about it.
That's creepy. That's creepy looking.
Just have to find one nudie break, and I'm gonna go and set up.
Just keeping an eye out for any vertebrates we might spot here as we make our way along this 100-metre transect. Once we complete the transect, we will start out collecting biological specimens and exploring the areas in a lot more detail.
Yeah, that's really where the fun begins, doesn't it, Corey? We can start looking for biological samples and looking for bright, colourful patterns and shells and interesting little critters. It's my favourite part of the day after these transects are done.
Absolutely. A lot of the time you've really got to put the RV on the ground and use the wonderful cameras that they have to zoom in and look for these smaller, sometimes cryptic mollusks or invertebrates rather. I'm a little bit mollusk biased because that's my background, but we are keeping an eye out for all the invertebrates, and it'd be nice to hopefully come across some corals,
especially some hard corals.
Yes, some hard corals rather. Yes, for Amy and Karen's various projects there. Hopefully we can get some records here in this true twilight area.
It is amazing how cryptic some of these species are, especially that spindle carry and how it blends perfectly into the fan it lives on. I think another perfect example is the pygmy seahorse.
Yes, absolutely.
Beautiful little nodules. That could be...
Where's pause? Is it a nudie? Or
could be a nudie.
Yeah.
Wait, pause?
Yeah.
That is.
Can we have a little pause there?
Pause?
Yeah.
Yep, we've got a nudie brand.
It's just on the right of the screen, centre. Yeah. It looks like a Philidia, yes.
Yeah.
Quite a large one. We might actually.
If it is, we'll just take a POI and...
I think we might just. Is it easy to grab it or?
Yeah.
Yeah, I think we might just grab it. It's...
Are we in the middle of the transit?
We are.
Almost.
We can...
You want to do the Niskins as well?
While we're here,
Should we do them?
we're fairly close to midpoint.
Yeah. Yeah, let's... We'll stop here then and
Take
find
a sample,
Niskins.
do the Niskins and
Yeah.
Niskins and collection. Thanks.
That sounds
Isaac.
good to me.
Certainly looks like a sea slug, doesn't it?
We'll just confirm it.
I'm just going to say pause.
Is it going to be a suction or am I going to pick it up?
So we're going for a institute.
I'm not sure now.
That's what I
Oh yeah.
Suction.
Probably suction we've been doing mostly, haven't we? With the sea
We
slugs?
picked one up yesterday. It just depends on the situation, really.
Narrator-
We're
We're
narrative.
sitting on some hard substrate, like a rock that you can pick up with it.
Yeah.
No, we picked them up.
Yeah, we picked them up just straight off the.
We're just landing at the moment. We're going to do Niskins and
Yeah.
So we've done a
Breathe,
pause,
narrator,
pause
breathe.
in the transect. Doing this.
Alright, it's all yours.
That wasn't the correct voice.
We've done a pause in the transect.
we had that one yet, haven't we?
Very excited about that. Let's get some crispy imagery.
I'll just censor that.
Couple of images, please, guys.
Hang on, wait. It's not crispy.
Fantastic.
Okay, this one.
Sample
in situ.
Let's just get a bit of a video. Look at that cute little dot on its nose. Oh my goodness, that is the cutest thing I've ever
Mm. There's some beautiful gills.
Early
Key
stages
message
narrative, but
waypoints.
what are we described, or it's too early to say?
Too early to say, but looks unusual.
Yes.
The problem with Chromatodorus species is that they can be quite variable and they can have a couple of different morphologies, and so only genetics really helps us understand which species is which in this genus. But looks very interesting to me.
What do you suggest for retrieving it? Do you want to do suction? Would that be the...
Yes.
Yeah,
Yep.
okay. I think we'll do suction, guys.
Alright, we're on one lead.
That is very, very cute.
Just trying to get an idea of size. What do you think?
Stand by.
Very small.
I can't see the dots at the moment, but
Quite small. I'd say it's probably about 7. Oh no, I take that back. It's quite big.
About 3 centimeters. Would you
3
say?
or 4 cent. Oh, it's a beast.
1, 2. Yeah, it's
I reckon
reasonable.
even 4 or 5 centimeters.
Okay.
4 centimeters.
I'm strong.
Yeah.
And when you're ready.
Ready?
Alright, nice and gentle.
You can start quite a bit. There you go.
Oh, well done. Expertly done.
There it
Beautiful.
goes.
Oh my god.
Fantastic.
How perfectly did that go?
So the next stage will just be checking the jars to see that the little guys made it in there.
Yeah, we definitely saw that one going there. So
That's
Yep.
definitely
Yeah.
on to jar
Yeah.
1.
Yeah.
Beautiful.
Jar
Thank you
one
guys.
confirmation from Chris. Yep, thank you.
Chronodoros. Safe and sound in jar 1.
Which jar? Jar 1.
Yeah, correct.
It was a jar 1?
Jar
Collection
one.
of
I think it was
Commodores.
jar
Would you
1.
like
I
to
think it
confirm?
was jar 1.
Can we
Yeah.
look to the right to make sure everything's there?
Okay, camera back. Coming in hot.
Stand by. Stand by.
I guess I'm catching up to do.
Sit down.
Oh, you want to get into that?
Amy, just so you're aware, we're about to fire in these things as well.
Alright.
So just sampling some water here for our eDNA analysis.
Yep, there's a go.
4's fired.
Pause as well.
Beautiful.
And 6.
Fantastic.
4, 5, 6, all successful.
4, 5,
Yes.
and 6. All
At
good.
100 meters.
Resume transect.
Just have a little
Initiated.
Alright, resuming transect.
Resuming transect.
There it is.
Oh, there's another one. Stop. Can we stop for
Oh,
a
I'll
second,
stop.
Great. Great spot.
Bridge, RV.
Yeah.
Hey,
Corey's on the fire
looks like
today
they're gonna
with
take
the
more
green
samples
spot is.
Initially.
Great to have him co-piling today. Eagle eyes
yeah,
here.
that's cool. We all stopped here.
Thank you.
I'm not sure it's the same. I only got a quick glance at it. Maybe our YouTube followers can have their comments come up about whether they think it's the same as the one we just collected. Of course, the one before had this, what I thought were characteristic black dots at the anterior and posterior end.
Yes, we'll see. We also had the yellow gills and chemosensory rhinophores.
Yes.
And she's back.
I'm gonna. And what's really spectacular about that is that it appears to be feeding on that sponge. We'll have a close look, but collecting that whole sponge and animal would be absolutely amazing. Oh, we're rarely so lucky to get several.
Is that like the one before?
Very lucky here today.
No, it had more
No,
orange
the
around
one
the
before
head.
had an orange beige rim.
Yep.
Yep,
As
okay.
well.
So
So
let's.
in situ shot of that.
Thank you,
Is this
Amy.
another Commodores?
It is.
So what do you recommend? You slurp the sea slug and then pick the sponge up after?
No, well, we could give it maybe an exploratory boop to see how easily that sponge will come up. If it's going to be annoying and scrapey, we may reassess and suction first and then scrape sponge. But if it comes off with it,
The whole thing into a quiver. So we can
yeah, yep,
sea slug and sponge into a quiver. Okay.
yep.
I think that'll fit.
So cool.
That is awesome.
Wow. Like
At
it never happens.
105 meters too.
Yep.
It's very symmetrical.
It's hilly.
Oh, it's doing a headstand. Okay, I think we should suction it, the nudie first. It's gonna let go.
We probably just need a piece of that sponge though. You want the whole lot?
Yes, it's not that big.
Okay.
Alright, so
Will you be
suction that.
investigating the types of sponges that the nudibranchs eat?
It's just very rare that we get
Feeding.
confirmation of feeding. I'll have a look for the feeding scar as well. But when they're sitting on it like that, You can
You
usually
need the
see
part
it.
that it's feeding on, or just a part of the sponge?
I would like the whole sponge. It's not that big, and we want to make sure our taxonomists have enough material to work with. But I think the slug will come off with the suction and leave the sponge there, and then I think
Yeah, I was just going to tear off a little piece of the sponge and then suck it up in the suction jar, but you need the whole thing.
yeah, no, I'd love the whole thing. Thank you.
Next, the sponge looks like an egg sac from an alien facehugger.
Just finishing the last image.
Okay. Like.
I just have
Okay, oh, that's good.
That's all we say.
Cool.
You're saying alien or alien? You're saying alien or aliens?
Is that the...
Well, that's just, well, you would, if you're not familiar with the facehugger egg, that's what it looks like there.
Well, amazing eyes.
One on Corey. Our slug spotter for today.
You ready?
No.
Co-captain Corey today.
Not Captain Corey.
Captain.
Captain Stick at the helm of the vessel today.
Big D stick.
Got it. There it goes.
Beautiful. Thank you
So,
very much, mate.
Two.
Jar 2, Chromodoros.
What jar was that?
Dose.
I think that was jar 2, yeah.
The feeling the sponge might come in pieces by the time I grab it. So
Negative Nancy over there.
I know, talk about doubting me from the jump.
Yeah, I know, right? Lean back here.
That's the prickly one we had first few days or
I
something.
like that one.
sounds related.
Okay, maybe right.
What's up?
Get back here.
He got lucky if he's alright.
You got a little food?
Yep.
Wow, looks
Moving.
something.
That was you. Okay. Thought it was me.
No, that was me trying to.
Forge out.
Okay.
Coming out.
I'll go out.
Standing by.
There you go.
Alright. Is he directly above it? That green one is actually inboard or aft, so you shouldn't have much of a problem.
Are those quiver lids on the roll?
Yep. I'll fix it. So I'll put 20's lid on this one. Correct? Okay.
Yep, that'll be 20.
Yeah, 20.
That's 20, yeah.
That's 19. I think should be center one.
And 21
20
should
over
be
there,
there,
21
and
to the
19
right.
should be
Oh yeah,
at the bottom.
So that's actually 20 that you just put that
yeah, it's
in.
actually 20.
Yeah. So I'll put this lid on what I just did.
And then 20 goes to
21.
where you just took off, and then 19 to where
I'll
you
just
took
leave
20.
that open, then I'll do the same thing.
So every night we take the quivers off from SuBastian and take in our samples, and then it's critical we take them out and wash them. But it's usually quite late in the night, and we put the lids back on them. Sometimes we don't get them in the right order, so
That is correct. I was
we're
in
just
charge
doing
of this.
a lid reshuffle, and Declan's put his hand up to say that it was his mistake last night?
Oh no, I was in bed asleep.
Oh, okay.
I knocked off early last night, so yeah, it would have been one of the other.
Yeah, it's probably Amy at the back.
Yeah, one of the other honours students.
Oh, I believe I was preoccupied. Thank you.
All I know is that I was given that rack by Corey.
Very interesting. A few accusations here.
No one sticking up their hand.
If we spread enough flame around, there's a snake.
Oh really? We got a sea snake.
Looks like we have a little visitor.
Oh, big snake.
Hit the snake button.
Snake.
Snake. So this looks like another olive, possibly.
Oh no, this is not an olive. This is a different one.
It looks different from. It has more patterning towards the ventral surface.
Yeah, he's down to the right. We can't see him. Well, if we fly away,
Did we manage to get a photo?
we'll take care of this first.
Yeah.
Fantastic.
I'll put a sample in that one.
Okay, and then 21 cover it. That's a 19 open.
Yeah.
Yep, that gets you flying in ahead.
Beautiful. Great work, Amy.
I think we're just going to move 21 over to the empty.
We're gonna leave that open for the next one, and then put it on, and then have 19
That's a
open
great
next.
idea.
Oh,
All over it.
smart.
You're
That's
welcome.
all J-Rod. I was as confused as everybody else.
Yeah, you can just get that in order tonight, please.
You're quite good at jigsaws, J-Rod? Yeah. I do love a 10,000 piece puzzle myself.
The ones with 3 pieces are pretty easy.
Yeah.
Alright, you ready?
Yeah, ready to go.
Continue on with the...
Thank you for that.
Yes, please. Yes, resume transect.
And here, confirmation.
Resuming transect.
There it is.
Thank you, Amy.
Before I pause transect.
Beautiful.
We'll take some good photographs.
Some good pictures of this guy.
What a beautiful snake. Lovely banding pattern down its dorsal surface.
Where's the little pamphlet at?
Yeah, where
I
is
haven't
that?
seen the snake pamphlet.
I'm not sure. I've seen that for a few days.
So yeah, it was yesterday. It was out. Whoops.
It may have made its way to the library, I'm not sure.
Seabed is still there, in case anybody's wondering.
Oh, it's nice.
Oh, look at that.
Very
Look at that.
inquisitive.
Wow.
Oh,
fantastic.
Oh, cool. That was awesome.
Yeah.
You don't want to go?
I don't want to go anymore. Yeah, we're just gonna get away from you because you're in a dangerous. He's in a dangerous
Is that
spot.
his 15 seconds of fame?
So he's in. I'm gonna let him
That
swim
snake's
off.
probably
so
going to go up to breathe
does it.
now. Get some air into his lungs before
I was trying to
go and dive
hopefully
back
see
down.
him somewhere. I don't want to get him into the thruster.
I'm sure we'll see plenty of other snakes on the journey here today.
Oh god, I'm scared.
Fingers crossed.
I was putting
Been
those
very lucky
cups
here.
on last night, and
Yeah.
Seeing
I was scared.
just about a snake every day.
We have, and at the start of the trip, we weren't really sure what we might encounter. We,
Interesting fact, I read an article that said in 1973, people were seeing on average 45 snakes a day.
Thank God I wasn't
And
alive.
what's the snake situation like
These people
between
live to tell
now
us.
and then? Is it unknown? Or?
Well, around 20 years ago, they mysteriously disappeared, and nobody has solved the mystery as to why.
There you go. Fascinating little snake facts there from Amy. Thank you.
Sea turtle's got an appetite.
What
Where is
depth
that
was that in?
snake?
What depth? Like, scuba diving depth.
I do it. 73.
But there's a lot. But apparently there's
73.
Scott Reef, which is just south of
Was that
here, apparently
somebody?
there's still heaps of these snakes
Yes.
there.
there
There
is
So it's just-
outside there.
Lots of snakes.
Yeah, so the disappearance seems to be kind of local to Philadelphia.
Yes, that'll be fine.
Yeah.
Alright. Starting again.
Right, finally resuming our transect after a few little distractions there. A couple little nudie breaks.
Some
And
fantastic distractions.
yeah, a fantastic little snake popped its head in for a bit, so...
Transect now.
Yeah, we're back on the transect now.
Talk about distractions. Lady Briggs,
Had a busy little period here mid-transect.
Boring old
We,
transects getting
we did stop and start it, so it will
Like
be
saturation
will
there?
be sort
So
of...
it'll just be transecting and just DIY.
Oh good, oh good. I would have stopped for both of those as well.
we figured Merida
I
might
mean, you got those guys in
be a little bit angry if we just strolled past two nudie breaks thanks. They're a lot harder to find later on if we just
Yeah,
point of
yeah,
interest.
she was pretty happy with those mini rings.
Yeah.
Some all happy dance as we speak.
Yeah, kept her happy for today,
Yeah.
which is the main thing. Fantastic.
Yeah, no, you guys are doing a great job. Getting some paperwork done in the background, so that's fantastic.
Good to hear.
Those are RV covered.
But yeah, this habitat's different again.
Right, yeah. These work with a lot of coverage.
I reckon we'll certainly have some fun snorkeling around here this afternoon.
Absolutely.
Does any of you guys want to go grab lunch?
The content.
I'd say it's smart.
I might go. I'll go in about 5
Yeah, make
minutes.
sure you do it.
Yeah, I quite enjoy it.
Well, we're getting pulled quite a bit on the boat right now, so it's gonna be a little funky for a second. Yeah, I'm getting pulled around.
I threw with him after that happened.
I know, but I think it's just that it was.
He did say the currents were changing.
Yeah.
It may go parallel to it, so with our eyes peeled for any interesting critters.
I'm gonna follow this break then.
Crab, yeah.
I've been prone to it. I've locked in. Shell.
Yeah, these nooks and crannies are very
Yeah,
good for
these-
hiding little
These would be
crustaceans.
good to
I think it's probably
ROV
this was coming right here. I think the blue's coming around here, so the current was just kind of swooping in around this.
Yeah, I'm just gonna hold him there. He's doing that jetting change.
Yeah.
And when you get a little bit closer, I can't do it.
And get around this, and it might get back to more smooth transition here.
Interesting encrusting sponges there on the top.
I hope everyone's enjoying the footage here from Ashmore Reef of the northwest of Western Australia, wherever you might be watching this live stream.
Very lucky to have such a diverse ecosystem and reef so close to our home base back in Perth.
Absolutely, it's a fantastic reef system where
What's that?
we're lucky enough to see it close up here as the ROV glides across this 100-metre transect.
Alright, we're past that little break, so we'll go back onto the flattish area.
I see there on the comments the snake appearance has generated quite a few comments there from the sea snake guys out there. Hopefully we can spot a few more the rest of the day.
I do love our sea snakes here on RV Falkor.
That's great shots of that last snake.
Mm.
Nice and close.
Came very close to our cameras, so we should have some good footage.
Does anybody in the audience, the snake people, do they have an idea on it?
Where is it, Robbie?
No ideas of yet, but if anyone has an idea of what that last snake might have been,
How much,
feel free to
more do you
comment
do on
and
the
we'll
heading change?
be happy to review that.
Certainly not an area I'm familiar with our sea snakes, so I won't put my hand up. Have a guess?
No, nor shall I.
Okay, can we start moving to the east as well while we do it?
Yeah, you can do that.
Okay, I'll give you a little waypoint.
Great,
My
thanks.
hero Luke.
Yes, Amy mentioned Scott Reef, which is another reef system further south of Ashmore, considerable distance. I dived there in probably 2010, so that's 10, 11 years ago.
Yeah, it's fine now.
And very healthy, very
Just
abundant
getting off of
sea
that
snake
little
populations.
curvature.
How was the coral there? Looking quite vibrant.
Yeah, it was amazing. But I can remember, sort of groups of sea snakes.
Oh, fantastic.
10, 15 sea snakes
No, I'm gonna fly right into that.
per dive.
It's amazing.
Yeah.
I'd love to be able to go over to Scott Reef or the Rolly Shoals one day and have a little squiz around, find some snakes. Be lucky enough to do that.
So are they quite inquisitive when you're diving with them?
Absolutely, yeah. Pretty much as you descend, as you do your descent, if they spot you, they make their way directly to you. That can be 50, 100 metres away.
They'll come over and
they will
check
come
you out.
and swim around you, around your mask, or under you.
Fantastic.
Have a bit of a lick, a bit of
a taste,
and
decide not
just
to
go
take a
about your
chocolate
business.
McClory.
Yeah. So I know that all the sea snakes that's been found in Australia are venomous, but they're not naturally aggressive animals, are they?
No.
So then,
absolutely.
unless you're really bothering them, they're not gonna harm you.
Yeah, haven't been many cases of snake fatalities in Australia, if at all.
Sea snake?
Sea snake.
Oh yeah, sea snakes specifically. Yeah, our land snakes in Australia are a little bit different.
Although I think there's still really low numbers of land snake
On the list of things that
fatalities
kill you in Australia, sea snakes are
very,
down
very
low.
far
How
down.
this heading is, and then go from there if that works.
Okay, sounds good.
I don't even think people really get land snake fatalities
No.
much
No.
anyway.
Very,
It's a
very
big
rare.
misconception that everybody dies of dangerous Australian animals, but they don't.
It's very safe here in Australia. Please come and visit.
They also offered that when that guy held that octopus.
Tourism WA.
You've only got crocodiles, sea
I think a lot of
snakes,
people are trying.
big spiders. It's totally safe.
So Amy, do you have any snakes at home?
Funny you asked me that. I do. So I have a pet snake. His name is Mr. Slivers, and he is a Woma python. So Woma pythons are endemic to the north and central, more desert-like areas of Western Australia. He's extremely beautiful, and I miss him a lot.
Fantastic.
Wow.
Yeah. Really beautiful pythons, those normal pythons.
He's very friendly. He stays on me while I study. We go for lots of play dates in the backyard. Likes to sort of slither around through the grass in the gardens.
Also, you take him for little walks. That's very
I
nice.
do, yeah.
Play dates with like rodents?
No. Although he did, I took him for a little play date at the back of, he babysat at the moment so that he's not all by himself. So at his auntie Caitlin's house. So I took him for a little wander out the back one day there, and he kept going to the side fence. It's like, why is he so curious about this fence? And he sort of went straight up to it. He kept beelining for it. Then I saw there was a little hole in the fence, and this ginger cat just had its head stuck through. So I thought, okay, playtime's over. We'll just go inside. Don't want to find out what would have happened.
See a very good point there from Insectoid 3 on YouTube, saying the vast majority of venomous snakes of all kinds aren't particularly aggressive since they don't want to waste the venom they need to hunt with.
Good point.
Very good
good point there, and thank you for that.
That's when they kind of just
All people who've had to use the
snap at you but don't actually bite.
Correct.
Yeah, snakes tend to be very timid, especially when you're hiking through the Australian bushland. They tend to just scurry away from you.
All my experience with land snakes in Western Australia, the venomous type, the only thing they want to do is just get away into the bush.
Except for sea snakes. They
Except
come
for sea
at you.
snakes,
Yeah.
yes.
Which are quite inquisitive.
And water moccasins in Texas.
Good point.
They come at you.
And most of the incidents on land are just accidental, probably accidentally stepping on a snake.
yeah.
It feels threatened. It sort of bites in defence, really.
You say the same thing about sharks?
Yeah,
all
no,
water crocs.
same thing.
They're all accidental.
They're all accidental.
Most of the time, yeah. It's just inquisitive curiosity behaviour.
They just have that like beat into y'all to keep the tourism going.
That's right.
Australia, that's the county.
It's not the land of deadly animals, it's the land of deadly accidents.
When we were in quarantine, they found that lady's foot.
Yeah.
next day they found a torso and it didn't belong to him. And I was like, oh, that's no good. That happened.
Well, in Detroit, that is fairly common.
Tiny little sea star in the central screen. Little tiny orange one. You're pointing at it.
Oh, yeah, this is funny.
We must have a pretty good collection of sea stars going now. Got a few nice ones yesterday.
I would say that would be one of our
Star. Would you say one of our star collections?
I would say it's one of our star collections.
I got an updated waypoint for your bridge if that heading's looking for.
Thank you.
Yeah, lots of soft corals, sponges.
I might just start. So this is Corey from the museum leaving now. I'll leave you in Declan's
Very
hands.
capable hands. Thank you, Corey, for
And
your
hopefully we have a
work.
successful remainder dive for the rest of the day. Thank you.
Enjoy your break. So we're over halfway of our 100-metre transect now, still just making our way to the end and keeping our eyes peeled for any biological samples that might be of interest, marking them later so we can come back and have a look.
Or stopping.
How long have you been doing the eDNA stuff?
How long have I been doing it? About 3 weeks. Yeah. So eDNA is completely new to me. What have you? You spot something?
Spotted something, but it's not moving, so I lost interest in it.
Probably a sponge.
Probably a sponge.
When in doubt.
When in doubt. Yeah, so it's been sort of a new area to me. I only actually just started studying at university as well, which is quite a coincidence that I got asked to then come on this trip and do some eDNA, because my primary interest is in coral. Lots of my my honours is on microbiomes and coral, and comparing those between thermal gradients to sort of identify heat tolerant species. So that could be applied to some sort of conservation management later down the line, but very preliminary sort of study in the Rolly Shoals. But yeah, very excited to be helping Karen with some of the population genetics on the coral as well. It's really cool. Fantastic opportunity I've been given.
Now you're hooked.
Now I'm hooked. Yeah, it's great to be able to learn something at uni, like in a coursework unit, eDNA, just this semester, and then start applying that in the field on a trip like this. It's been really, really worthwhile. How long have you been driving submarines, J-Rod?
About 10 years.
10 years.
Sebastian for 4.
What sort of submarines were you driving before?
Different brands,
Different-
mainly for oil and gas.
Yeah. All remotely operated?
Yeah, no, I've never done any of the
Yeah.
submersibles. I would do it in a heartbeat.
I've seen too many submarine catastrophe movies to want to go on one, I think. remotely operated the way to go. You can stay dry and air-conned and just point at things on a screen.
If you think about it, I mean, like, it would also be like you would never fly because you've seen so many movies about plane crashes.
Yeah.
The odds of you actually
Yeah,
crashing.
actually happening is very low.
It's even less for a manned sportsman. So.
Yeah.
The safeties and everything that they have is pretty rigorous. Like the training that the pilots have to go through is like
very
extensive.
Yeah,
Very
extensive.
Yeah.
Who knows? Maybe one day.
Ooh, that's a big.
That's interesting.
What's for lunch today?
I have no idea.
Yeah, it's just a game like watching the score instead of it shows you perspective.
Oh, so it's actually like reflecting
The light, yeah. It's more like kind of like a football match. Like it just shows you know, against all the shots.
Yeah,
That's well.
That's right.
That's
That kind of thing. Something to identification.
Yeah. ¿Qué pasó, chico?
Monday.
What you want in this one or that one?
Rest of the
Oh, I'm sorry. I figured you go first since you've been here long.
Not a problem.
I can sit there until he gets up.
No, you're fine. Just steadily moving left. Sometimes front and back, but yeah.
So these three groups were all on bed. So this one's open right now. And number 20 is actually where 19 is supposed to be. Lid number 19 is sitting right here. So we put a sample in there, take 20, one, there, that's it. You'll have that one ready to go. They've been used, it's just all three of these lids were put on backwards. So they had, I just need to they had lid 1 sitting on that one. When I went to the sample there, I took the lid here, there. This lid that's sitting here is actually 19. This says 20, so put a sample in this one, that's that lid. And then 19.
I just said this is so
Fine, dude. How about yourself? I'll finish this. We're taking like 2 more minutes. Yeah.
lovely.
Oh boy.
Let me take your own.
Yeah, if you want to. It's all yours. If it's something that you would enjoy, I'll be more than happy to let you do it. Because you know me, I'm a giver by nature. I'm also a hoarder by nature.
What do you hoard?
My money when you ask me for it.
Think of the kids.
If you want to send them to me for the summer, that's fine. But I'm not going to sneak each one. I don't know what you guys would do with it. Probably buy snakes and stuff. unflattering stickers on the back of your vehicles. Yeah. We didn't really do much marks to go back because we just stopped and picked up what we liked. That's how we do it. There's also in box 3 on the right, there's a sea star who does not want to be there and tries to escape. So if you do have to open that, just be wary. Sitting there.
Is it a big one?
It's fairly big.
Bigger than that 2 kilogram one?
No, it's nowhere near that big. It's a little bigger than my hand.
Yeah.
But it was on the edge, like the top edge whenever we opened it earlier.
He wants out.
Yeah. They were going to bring their heading around due east, but I don't know if they're comfortable there or we're just waiting for us to finish or what the deal is with them.
And what do you reckon about that? Slightly comes to the right of screen? Yeah.
If you go back to the top of this transect where we started, it was very, very, very full of stuff.
Substrate's becoming a bit more sandy again. We get towards the end of our 100-meter transect.
Are we nearing the end?
Yeah, we're coming towards the end.
We're going forward. Why did I just better go back to the nice rocky platforms?
Yeah. Some
Hanging.
beautiful rocky substrate earlier with lots of little nooks and crannies for our biological samples to hide in.
So if you look right there, that dark crevice looking thing.
Yeah, that looks great.
I would recommend that.
I think we'll be subbing out soon enough. Karen will come back in so we can grab lunch.
I'm gonna do that myself.
Yeah.
Declan could go away to the end of the transect. Or wait for Karen to bounce back this time.
Alright, thanks for tuning in. I'm going to head off and grab my lunch now, and Chloe is going to be taking over for a little bit. So, thank you very much for watching this morning. We're coming towards the end of our 100-meter transect now, so I'm sure Chloe will take you through the biological sampling, and we'll have a little squeeze around and look for some cool stuff.
10 days, yeah.
Thank you all. See you later.
Hey Chris, I heard you're going to run a quiz night.
Fine. Doing a quiz night?
Yes, Chris. Please.
Yeah. All German philosophy and Bible questions.
Hey, I got an A in RE.
Alright, that's pretty much the end of said transect.
Alright, and do transect.
Yeah.
Okay.
Confirmation. Cheers, Chris. Yep. Good. My finger. Chris drifting away.
Oh, thank you.
Drifting away.
How's lunch today?
Don't want to be parched. Right now, good. I'd go for the soup. Yep. Soup is top notch.
Soup? Yeah. What kind of soup?
It's Thai green curry kind of soup.
Alright.
Right then, where would you like to go?
We're just hanging out, I think.
So I think we're actually gonna go back a bit because this wasn't. There was lots of really cool rocky formations.
Okay.
That Chris is gonna take us to, but he's obviously.
So you want to go back up north then, eh?
Yeah.
Back
Right.
to where we came from. I think there was some interesting ground.
Head to
Thanks, Russ.
breathe.
He did point somewhere.
ROV.
I just. I didn't realize it was
Cool, so we just had a little changeover there, guys. For those of you that are just tuning in, hi, my name's Chloe. I am here as a researcher on board the RV Falkor, and I am primarily a honours student at the University of Western Australia, but I'm working with Dr. Narrator Wilson at the Western Australian Museum looking at sea stars.
So Chloe, what is your favourite thing about sea stars?
Apparently this is an interview. We're turning this into a little Q&A. My favourite thing about sea stars is, I don't know, there's so much to love. There's so much to love. I think probably after being on this trip, my favourite thing about sea stars is just the diversity that you see in them. We're seeing on every transect that we do at every location a different kind of sea star, and they're all different colours and shapes, and they've all got somewhere between 4 and 8 arms. It's ridiculous. But yeah, I think it's just cool. Everything's very new for me all the time.
What are the kind of sea stars that you're looking at for your project?
So I am looking at the family Pterastidae. So they're primarily deep water species, but they're also found in Antarctica, and there is one lonely little species that's found in the more shallower regions on the continental shelves, and that, oh, hello mum. That one's called Eurytesta insignis, and we actually found one yesterday that was very exciting. Hi Kat, nice to see you're still with us. Thanks for sticking around so long.
Do the Pterastidae have any like distinguishing features?
They kind of, I think everyone's quite familiar with cushion stars, which are kind of the ones that have quite a thick body, so they're a little bit chubby, is most of the time how you distinguish them. And they've also got a very prominent osculum, which is that little hole in the centre. So like when we had it out yesterday, we had it sitting in the lab and it was kind of opening and shutting this little osculum, which is really cool. So they're quite fun to spot. Thanks, Amy.
Are there any shipwrecks located on Ashmore?
Are there?
I don't know about any shipwrecks on Ashmore, but we did find an anchor the other day that looked extremely old because it had a lot of growth on it. So I think we're sending photos of that to the Maritime to check that
Pretty
out.
cool. Thanks, Amy. Hi, Lisa. How you going? Nice to see you back. I was wondering when you were going to turn up. Cool.
Chloe,
So,
what's been the coolest thing you've seen so far?
the coolest thing I've seen so far. That's tough. I feel like, again, it's just that this is not necessarily just a first for me, but a first for a lot of people in the fact that we don't explore Mesophotic reefs quite as often. So just so many of the things that we're seeing are new and weird and super awesome. So I don't know, I don't think I can pick a favourite. I am biased in the fact that I found one that I'm studying. So I'm like, oh yes, the sea stuff. What about you?
The sharks.
The sharks.
The sharks.
You've seen some good ones. I think I've seen like one wide tip, and I'll take it.
Sharks are pretty cool. And all the fishes.
I was telling the ship to shore kids today about the conspiracy theory about the shark tearing out the cables on the ROV.
It's not a theory.
Not a theory.
It's not.
100% true.
100% fact.
Oh really? What's the evidence? Prove it.
Well, we don't. Okay, well.
But there's no evidence.
We'll do a cutout.
Well, there
of
was
the
no
shark
cable
on
attached to the
cardboard.
vehicle. That was fairly evident.
So there was a person down there ripping
hold
it
up
out.
in a court of law.
Well,
Well,
no,
it
but a court of law.
depends who you are.
The evidence is circumstantial.
God, I think
Thanks,
it's more
Judge
fun.
Amy.
Well, it's more likely
Thanks,
to be
Judge.
a shark rather than a nautilus that came out. It's just, you know,
Look, my money's on an octopus.
octopus.
I'm cracking.
A sneaky little octopus. A kraken for sure.
Cracking, for sure.
Mysterious cracker.
Maybe a
I think that's right
mermaid.
behind, that's,
That's the weird thing.
Yeah.
You would think there would.
because the sharks don't have little scaly bits, you know. That's what I'm thinking.
Snake behind you.
Snake.
There's a snake behind us, guys.
Yeah.
Don't know what kind. Ribbon snake.
Ribbon reef snake. That's the one.
Just gonna ask my wonderful co-worker Amy to come and sit with me if she'd like to. Yeah, Jason's just said, I think what he's implying is that his favourite thing so far has been a short-nosed sea snake, and that was really exciting for us. For those of you that haven't seen the various news stories, there was a while there, 20 years or so ago, where we weren't seeing many sea snakes on Ashmore Reef. They used to be very plentiful here. And this is kind of the first time that we're seeing a few of them coming back, and it's really exciting.
Not for crystal.
Right.
Help me find some cool things. I found a cowrie shell, like a spindle cowrie. And
I saw it. It was the first thing I tuned into, and it was walking across the floor.
And a sea cuke. And that's about it so far.
The demonic smile.
And Dr. Karen Miller is back in the house.
Take it away, Karen.
now you guys are fleeing. Whose cup is that?
That is Declan's.
She's here. I'll take it downstairs.
Do you want me to find this in the fun?
How was lunch?
Lunch was delicious as always. Yeah.
Now you need a little nap.
No, I'll sit back and watch some telly, it's alright.
We got some really great photos of the snake this morning.
Yeah, I was in the other room and I did see it on the screen, so yeah, that was cool.
Nice. Close up fish.
So
Just
good.
heading back up the line again for you, so you can.
Oh, okay. Was there some waypoints that were marked, or we're just starfling?
There were some two starfish marked sort of before we even got to the transect.
Yeah.
But I think
Oh,
we're
okay.
going backwards, so we can probably just slow
Oh yeah,
down.
we can just cruise back. Right, well we've just changed the guard. We're all shuffling for lunch. Thanks to a wonderful team of students who've been keeping the narration going and keeping you informed for the last few hours while narrator and I have been busy doing some stuff in the background. I'm Dr. Karen Miller from the Australian Institute of Marine Science, and yeah, I'll keep you company for the next little wee while on the dive. And we have the capable Russ and Cody piloting for us this afternoon. Oh, can I just have a little closer look at that on the way past? It's probably a sponge, but
Karen, we asked the girls already. What's the coolest thing you've seen so far on this expedition?
oh, I can't name one thing. Everything is
Exactly
cool.
what they say. Okay, what was your most favorite?
Oh,
Top 10 or top 2? So top 2. Like you're just
so it's interesting because
flabbergasted.
every evening at the end of the day, I sit and make a whole pile of observations and write notes about what we did and what we found. And I've always written at the end of that, I've written a little highlight on each day as to what my highlight was. But as to which was the best of all those highlights, oh, gee, that's a tough one.
You just sat there and you're like, whoa, I did not expect this to be here.
Yeah, so kind of put that question to me as well about what have I found that surprised me, and at some level a lot of it hasn't surprised me because I've spent a lot of time staring at images of the seafloor around the northwest shelf, so I had a fairly good concept of what was generally going to be there. I guess some of the exciting things have been like really seeing how different as we've moved all the way around Ashmore, how different it was in every single place we went to, even though we're still only, operating on the scale of kilometres kind of thing. So that variation, the spatial variation and the small scale of that has been pretty cool. So yeah, and I think there was one dive we did, I'm trying to remember where it was now, it must have been off the northwestern end where there was just a stack load of different black corals. That was pretty cool. Yeah. I have a little fondness for black corals because I spent some time studying them in New Zealand when I was a postdoc.
Get some shots of that one.
Now, what were we looking at? It is just a sponge. Just a sponge. It's a very nice sponge. Probably good to just have a little snaffle around on the fine scale to see what else is down here. Although this is definitely one of my fun things, like really getting in close and looking up at stuff. I think that's, you know, that's what we've never been able to do before. So we've always been able to do the high level, broad scale stuff. So yeah, big picture patterns, got quite a lot of information on that. But this level of detail, yeah, you know, can't beat getting right down there. Even though those are the bits that are challenging for you guys to pick up.
That makes it fun. Makes it fun. Keeps us on our toes.
And so, Cody, did somebody ask you that question? What's the best thing you've seen this trip, given you must have seen so much being an ROV pilot?
Would say the sharks, but to be biased, sharks are fun to look at. Pretty cool stem we got yesterday. That was cool. That was neat. That stem.
Oh,
that we got
yep.
yesterday, that was moving pretty fast. I don't think I've seen one move that fast.
Yeah, okay.
That was pretty neat. Shark. Speaking of shark, there's a shark behind us.
Oh, sneaking up. Must have heard we were talking about it.
A little shark here to burn.
A couple of them.
Yeah.
I'm just seeing a few sharks out the behind us, effectively. Oh yeah, I can just see the shadow.
Just so it takes a minute of the light.
Is that weird doing that there? More stuff to the shark.
See, we're here
Right now.
John.
John.
Isn't it? Was it? Oh,
There he is. He's back. See? Maybe his name is John.
The shark named John.
Could be a good book.
Not how much you gave me, buddy.
GPS about us today.
That big block of cheese. There he is.
Speaking of little tiny things you wouldn't see normally, look at those little guys hanging in under that little crevice. I think they look like stylosterid corals to me, a type of hydrocoral. They usually like to hang out under overhangs. Like little tiny ones. Okay, we can continue meandering, thank you.
Hello, John. Go in. Pretty John.
Alright, Rich.
Good afternoon, Eric. We got an updated waypoint for you.
Thank you, sir.
You're welcome, kind sir.
enough samples every now and then. So just keep updating.
Alright, thanks.
You're welcome.
So Russ, did you get to answer that question about what you think's been the most impressive thing you've seen so far this trip?
Partial to Nautilus. to be
Ah,
honest.
yeah. They're pretty hard to beat. They are cool.
I really like them. I just don't know what's something about it.
Have we seen one today? No, not
Not
yet.
like today, you know.
No.
You remember one for a while.
I do.
It's
Just
been
like
a-
Nautilus. It's just
Oh, we did see one yesterday. But it was just sort of in the background. We couldn't see it in all its glory.
such an old, like, 'cause you gotta remember, it's basically based on a prehistoric style.
Mm.
I don't know.
The mechanisms of how it works.
And the spiral, of course, is a physics reason, which is really interesting.
It reminds me of a football.
Yeah.
You know something?
Yeah, they certainly don't look like something that should be able to swim at all, really, do
They're
they?
actually surprisingly fast.
Yeah.
When you try to chase them. Like,
About you, mate? Can't say Cody. What's your favorite thing? Can't say me.
I think it's when we sit on the seabed and you just zoom in and you see so much more life than what you would see otherwise in the first view. So that there's so much more behind it on that little square foot or square meter.
Yeah, especially in those deep areas where it superficially looks really quite barren when you look up from above, and then you zoom in and it's just teeming with life and colours and just everything's really small.
What I find cool is when we took suction samplers of just the sediment,
Oh yeah.
and then when you get the jar and you see all these small little critters just swimming around it, little brittle stars.
That is a seriously funky sponge. Is that the one you guys swam around before? The texture on it is great.
yeah, it's amazing.
Oh, you guys probably weren't driving when they were doing it before.
Shit started to move, Russell.
Time to play. Can we sit down?
Yeah, let's have a little look around here.
Sit down here.
See who's hiding under that sponge.
There we go. Right there, good to go. It's an interesting thing in front as well.
I think there's another sponge. Let's just have a closer look.
Always
It's a very
fascinating
strange
colours.
shape, isn't it?
Yeah.
Jump this back.
Yeah, it is a piece of sponge, but it's just an interesting shape. Oops, another nudie brain. Over and have a look a bit closer, I think, in a minute.
Yeah.
Let's finish. Around here. It was our head shimmer.
That top layer looked like it.
I'll take it going for it.
The good thing is, I think the bottom piece is done.
Yeah, you've got two stuff. So.
I don't know why it's normal size, so it's not extruding, because it's like,
This needs to be. Yeah, no, it's all
Just wanna get one of that sand out.
Yes, please. It's just say yes.
Is it bougie? Fancy?
It's quite it's posh.
What kind you got? Good flavors.
That was good. Thank you.
You ever in LaCroix?
Yes, sir.
LaCroix.
Croy.
LACR-IX.
We can sing Croy.
It's not bad.
All right, no. Everyone's getting mixed like that.
This is true.
Sip coffee. Have you tried it?
Bad idea. Instant regret.
This wasn't the friend we're looking for.
So there's our little friend Nudie Brain.
Okay, I shall manoeuvre my way
pop
around
over
and
there
sit on the other
and-
side, and that'll
Yeah,
save us from
thank
getting
you.
crush and then all that kind of stuff.
I got a pause right now, but I'm just gonna board the print so it cools down.
Yeah, that's fine. Just double check where it is again. I think it's sitting there.
Somewhere in there. Took my eye off the ball for a second. So we're looking at it from that direction.
Yeah, I'm sure it was in this area. Ah, okay.
Oh, is that? Yeah, okay. It seemed much bigger when we were looking at it.
I know.
It's actually a different one. So let's just have a look at that one and then we'll try and find the other one.
Okay.
Shimmer, shimmer, shimmer.
Yeah. Which one you have? Tangerine? Or
It might be better if we get a bit closer to that little
Yeah.
guy. Except that's not the one that I was looking at before, but we might find him on the way as well.
come on this side.
Keep an eye on this one. Now I think
Just gotta get where a land is. Okay.
thank you.
place now.
It's very similar to the one that they collected this morning. I'd rather put it as close as I can get to it. So, in the zone. There it is, just a moment. See if she might be another one of these.
Very easy.
See if I can spot the original one.
You know what? It's probably behind one of those mounds.
Yeah, we're probably just at the wrong angle to see it, and I missed it. She says, hell yeah. Hell yeah, okay. So let's get this one and then we'll go search
Yeah,
for the other
that's
one.
fine.
Would you like
Yeah, suction.
suction it, right?
Oh, I think that's going to be our only option for this little guy.
Let's do it.
He is on the sponge though, so I'm sure she wants the sponge as well.
Okay. Well, if that's the case,
Do you want to pick? Do you reckon you can pick the
grab
whole
the
thing up?
sponge, maybe.
As long as we don't lose the nudie in the process.
Yeah, because they don't really stick that hard to the.
No, I suggest suck the nudie off and then we'll pick the sponge up separately.
Yeah.
Just to be on
And
the
maybe
safe
if I can break off a piece of the sponge with the suction, break them all up at once.
As long as it, yeah.
Let's
yeah. See how you
we'll
go.
play it by ear.
Yeah, we'll just go easy on it.
It's going into jar 3, I'm guessing.
Yes.
Yes.
Jar 3 is selected. So that's basically. Sorry.
Yeah.
Let's go zoom back in again. Okay. Zoom in.
Sit down just a
It's
little.
just coming down.
Perfect.
Yeah. See when?
Yeah. Let's get it.
Because this is the time that it will stick. Oh. And got a bit of the sponge as well. Excellent.
And it goes.
Let's let it run for a little bit.
Yeah. All I'm gonna do is I'm gonna give it a little shake.
We'll power. Okay, just gonna move the camera around.
Okay. I'm gonna hold the suction. For the time being.
Check.
I wasn't. What is in 4?
3 in theory.
These krill. Anyway, back for that krill this morning.
Oh, there it is. That would be the sponge.
Find a little guy on there.
Let me see if the nudie's still attached to it though. Not quite.
There he is. See, he's right. Have you looked
Oh
at-
yes, yes, I can see him crawling
He's
along.
crawling around wondering what happened to himself. He's just a little
It's like, I'm really dizzy now. Perfect, thank you.
Let's re-index this back.
That one Sharpie was yeah, that one.
What I'll do is I'll move round to that side, then we can look at everything. on that
Okay,
angle.
yeah, that's a good idea.
And then we'll, get an idea. I can see it as well. Okay, I thought I saw over here. I'm looking for the set camera, you see, so I'm looking at that dark back.
Oh, okay.
Okay, are you free to
Okay,
explore?
I'll see what I can find. Now, oh, that's right. So hang on, where's the reference sponge? The reference sponge is
That's
there.
right there, yeah.
And so we were actually looking from that side before, weren't we? Up that
Well,
way.
okay, we're kind of pointing this way, we're kind of looking through there.
Oh, that way. Got you. I'm with you. Yep. So yes, he should be in this
In this
direction.
area somewhere.
Other way.
Thank you. Thank you.
Oops, wrong way. No. We might have to go back to the original position.
Yeah, okay.
Obviously, just the angle that we're at, he's obviously just tucked behind something.
Right, we'll go another one.
Let's try that, and I'll keep a better eye on the location when we move. So I think we were, looking from, yes, round there, that's right.
So what we'll do is we'll start off here, because we picked a spot that we didn't want to land on any corals, see, so this one here is fairly open. They're also a little bit closer, so.
Okay. It's got to be up in here somewhere.
Okay, you're good.
Thank you. Come on, little nudie, where are you hiding?
He's wandered off.
Aha, there it is.
Elusive little creature.
It has actually shuffled around a bit. It's a fast moving one.
Spikes. RV camouflage version.
Got the
That's
photo.
good.
Thank you. I'm pretty sure she'll want it. She's never turned one down yet, has she?
Right then. Are we to put that in the same jar, or do you like to put it in a separate
No,
jar?
I'd put it in a separate one. That could almost be pick-upable, or do you reckon we should slurp him? How big is he? He's a... Oops, wrong way.
It's probably going to be easier to slurp it just because of the size, without damaging him.
Yeah, I guess he's only a couple of centimetres, isn't he?
Yeah, he's not
Slurp away now.
No.
Ready to go for it?
Yeah.
Alright. And I'll try to get a piece of what he's on too.
Yes, it does look like it's on a bit of sponge, although he's definitely moving rather than sitting around eating.
Alright, zoom it out for you.
It's going to be jar 4.
Jar 4 is selected.
I'm ready.
Okay, coming on.
Right.
He's on a little rock.
Yeah, but just a nudie on his own will be fine, I'm sure. Oh, there he
He's
is.
in there.
He's in.
He's inside his new home. Hope he enjoys it.
Get a nice view from up there anyway.
Oh, we get to see all the other people that's been.
All the other dudes. All the buddies.
Yeah, having a little nudie party up there.
Oh. Right then. Good job.
You can go and back up. Oh no, you saw him going, right?
Yeah, he's good. Alright, secure.
Okay.
Would you like to?
Continue on our journey?
Yeah, let's go for a drive.
So we haven't. Yeah, okay. So yeah, we might once we finish this transect, just go down the edge of which
Okay.
that at some point. So we can start a bit there. Although you would have transited back up across
Yeah,
that.
transect's
would-
done.
Yeah. No. I mean in terms of between the two transects, when you went across that slope,
Yeah,
if you
but
like.
it was higher up. It was higher up the chart, so who knows? It doesn't look as though it was quite as significant. Because where we crossed it, it wasn't really.
Oh, okay. Oh well, we'll keep exploring this in the short term.
Yeah. amazing with the colours of the sponges though, isn't it? It's like you look at them, it's like there's such a wide range of shades.
Shaped sizes, there's blues, pinks, yellows, browns, you name it. Sponges do it. I admire anyone who's brave enough to become a sponge taxonomist. Particularly in this part of the world, it is actually a bit of a biodiversity hotspot for sponges,
Right.
the Northwest Shelf.
So what's when they preserve them in the museums? How do they preserve the sponge? How is it? 'Cause it's quite fragile, isn't it?
Depends on the type of sponge. Some of them are actually really quite hard.
Because I remember we've seen those sponges that got it's like glass shards.
Yeah, they're the glass sponges.
Yeah, and
yes.
they're ridiculously strong, actually.
So typically, I think they preserve them in ethanol,
Right.
or freeze them if they're especially large ones. But the challenge with identifying them is that you've got to look at all those. You're talking about those sharp glass-like things in the glass sponges, the spicules. Well, the glass sponges have silica-based ones, but the other types of sponges have calcium-based ones. But you've got to basically take really thin sections of the sponge and look at the detail on those spicules to really identify and distinguish between the species. So it's a lot of hard work, and there's such a diversity of sponges. It's more than many people's lifetime to actually go through them all and understand what they all are.
Hi, Eva.
Hello, Eva. What's that?
It's just a smidgen. How's it going?
Good. How did you go with your calibration this
Well,
morning?
I won't know until later, but I was, conditions could have been better, but I like the area that we found, and they drove did a great job in describing it.
Excellent. Good.
What are we looking for now? We'll come back
Anything
off
and
the slope.
everything. We're just sort of retracing the transect and
Oh, if you've got a
just
waypoint
snaffling
there,
around.
Got an update waypoint for you, buddy.
Oh, I see. Oh, that's where
We're just snaffling around.
Okay.
Haribo cow.
Eagle eyes.
Shoot sponge for these people.
Should we pop over there in front of
Sure,
some
go.
of these fans and we'll have a little look at them?
If you want, I can sit down round about in the centre here,
Oh yeah?
and then you can have a little look.
Capture a few from there. And just turn slightly so that we're, oh no, that's actually okay for that one.
Coming round.
Yeah. It looks like a giant nose.
It does. Like a monster nose.
A dog nose, no, a little dog nose. Like my puppy.
Fairly large dog.
Maybe it's a luck dragon's nose.
Could be. Right on the edge here. I'm just going to have to hold it manually. If you want to take the camera and you can have a look.
Oh,
at it.
okay.
Oops.
Anything that looks.
Take me through it a little bit more, too.
Should be okay once she's balanced. Nope, she's not doing it.
She's not happy? Surely it's a he. definitely love dragon nose, that one.
As soon as you let off,
And we're off again.
it's just the angle we're at.
There's not.
That's better.
Better? Thank you.
Birthdays after tomorrow.
There's not a simple flat surface on that thing anywhere. No. You want me to remodel it? So yes.
just sunk it through the bed, but if you want to put a flat base on it, go for it.
There it is, just like.
That's just what he gave us. I just sunk the model through the bed that gives you a flat base.
What is it? Roll sponge?
Okay.
Just trying to work out what that is. It does. I think we might have seen it. It's near the storm boy, though. Can you go and ask her for this and see whether it's something important? Oh, it moved.
Yeah.
Oh, thanks. Good spot.
Try to get something different light.
Well, it does look like it's hot. What's that? No, we can't. It's not even like it. We might need, that's full zoom, we might need to get a wee bit closer. Suction, I think. Can we just maybe get a little bit closer to that and get in the position? And there's a little tiny snail on there that we will try and...
We'll give it a shot. Pretty close, actually.
It's just, I think, probably the angle thing. Maybe we
Yeah,
just need to
sure.
rotate around.
Come in here. Too close for you. Right.
No. Probably if I turn the camera around for example, 2. Is there any way we can just twist a bit, or is there something in our way? Other than a luck dragon's nose.
There's a coral right on the front porch. You see how I went over it.
Oh, yeah.
Is that angle?
Let me just zoom in and see if I can still see it.
Hold on. Stabilize. Okay.
I think we've needs to be around. Oh, hang on, is that it there? There it is. Still at full zoom.
Oh, okay.
No, that wasn't it. I think it's sort of around there.
Okay, let's go around the back.
We can get so that we're front onto it again, if you like.
It's quite close there.
It's just leaning over slightly the wrong way now, isn't it?
Okay. Okay, let's move.
Just testing your small scale maneuvering skills.
That's,
Oh, that's probably a better angle. Ah, yeah, I can see it now. Yeah, it's just in there, which is incredibly difficult.
you can run the suction sampler down there and
Yeah,
hopefully get it
I
off.
think that's about our best option, really.
Because it's, I mean, it's fairly close to easy access.
You want to
gonna be
clip
a-
it?
It's an option
Snake.
too.
I have
We
no
got a
idea
snake
how
right
well
next
it.
to
I
us.
have no idea how well it's going to stick.
There's a snake in the front.
Sorry.
Snake.
Oh, sea snake. Where's he gone? Around there.
Gone.
It's gone.
Yeah.
Come back, little one.
He was moving fast. Good place to go. People to see. Fish to eat.
No. I was hoping you might come back, but no. Okay. Back to the coral. I'm going to find it again.
Oh,
It's,
is that-
it's in behind there.
Is that one? No,
It's. I think it's that in behind there. You can just see it. It's not going to make a very good in situ photo though. Did you get it from the other angle by any chance?
We've got some ice maps, but nothing fairly.
Yeah.
Oh, we can move back and try and.
Oh, I see him now.
See him? He's just
Yeah.
in there.
Yeah, it looks like he's moving down now.
Oh, yeah.
You can see the little white ball in the middle.
Right there.
Yep.
Yeah, that's the one.
See it's
Yeah,
little.
it seems to be moving about a bit, which is why I'm a bit worried it might fall off if we try and
Yeah.
cut a branch. So I think suction might be the best option.
Okay.
If you can get that angle.
You want to try to go for it, or
Yeah, unless you want to reposition, but it's like it's really because he's kind of half on the on the underside too, if you like. How confident are you feeling, Cody?
confident.
Your challenge, therefore, should you choose to accept
I
it.
accept it. I accept.
Oh, he's coming a bit better into view now. Can you grab that photo now? Yeah. Not that.
There he is. Yeah, he is moving around, isn't he?
Yeah. I might just try and put him in the centre, just for the photo effect. Yeah. Very well camouflaged. Okay, there.
Yeah.
Got it?
Yeah.
Okay.
Okay, can we blow the camera so that?
Yes, it's all yours.
When we're ready. we come in from the top?
Thinking about that, but I want to move this.
Right, we're gonna put it on anyway.
Oh, that's a bit, actually that's a better shot.
Okay, look, I'll hold this right here. Go ahead and go for your shot. Let you see him.
Hang on, let me just put him in the centre. Oh, the other centre.
Do a quick zoom out, and then slowly zoom in on him. I'll move this out the way so the camera doesn't pick it up. You got suction on right?
That's a better photo.
This is going into
Okay.
Alright. You
Do
ready?
you want me to zoom out again? Or you
No,
fine?
you're fine there.
Okay.
You're okay. Slowly move this into the position.
Yeah, maybe he's going to stick on better than we thought. Maybe he will just come off if we cut a piece.
You might, because that's a.
He seems to be holding on pretty tight.
Yeah, he's definitely.
Okay, plan B. Plan B. Is it okay to grab right where that top finger is? Right there?
Do you want to, yeah, maybe cut down here or something?
Yeah.
Yeah.
Just keep an eye on him just in case he does fall.
Yeah.
I'll go on there and grab this section.
About halfway in the blade, and I it's sharp as a bit. Try that.
Yeah.
Yeah.
Where do you think that?
We're certainly not going to fall off.
There you go. Done. Nice job. Right, let's get the coach out.
Follow, guy.
Okay, you've got one open already.
Is there? Hold that right there.
Okay, all held. Customs. If you get set, set forward.
There.
Zoom in, make sure you stop it.
Yep. And he's in.
Woohoo!
I'm gonna put the coral in too.
Yes, please. No, those are the two I've collected. I just collected this.
And it goes.
That might be one. Yeah, I think I know which one that was. It was a white sponge. It was one of the similar to that. This purple one. But it's not on this one. Ooh, lovely, thank you, gentlemen.
Okay.
Well,
Right, where were we? Should we keep just looking around this spot?
Yep, camera's all yours.
Thank you.
Knock it out.
So, what's before?
Hang on, I'm in the tools. Okay.
So, an off-your-road party happening on this coral.
thought it looked fairly familiar.
Okay, shall we continue our
Certainly.
afternoon stroll? we sort of meander just down that slope,
So we go.
get a little bit of variety.
Updated waypoint, Eric.
Oh, sorry, I just want to look at that urchin as we fly past because it's got so many fish hanging out in it, and it's a great colour. Oops.
Let me fly in there. I'll close it.
Okay. That definitely requires a photo, doesn't it? Definitely.
Okay, that's me stable.
Thank you. Those fish do not look that convinced about us. That's so cool. Little fish hiding in the urchin's spines. Pretty psychedelic urchin as well.
It's pretty neat.
It is, isn't it? Oh, since we're here, I'll have another look around.
Yes. $5. $5 a day.
Did you see the 2 lines up here?
No.
That on it?
No.
So I can take it off?
Yeah. Last people today. This is a Just get some grub screws for the nice thing for those bicycle connectors. In order to last the car. Yeah. Yeah.
Some hard ones.
Yeah. Get a nice ATM2 stainless. Tie.
What tons of Alex for it?
That would be a good plan. Yes, we can break more drill bits. That'd be good.
Well, just get out of the holes.
He doesn't use them already, so what else is it? Big company.
Some for a drill bit yesterday and just go working out one.
Alrighty. Thank
Super.
you.
Just keep going down then.
Yeah.
See what we can find.
Oh, there's
There you
one
go
of your
again.
friends.
He's munching away on that one.
That's a big sea star. That's starfish. I think he just got dropped.
I'm guessing I don't need.
Looks like he's at one too many.
Everyone will wake up and look at this food, right?
Yeah. Can't find about that, right?
It's like somebody just took it off and threw it over the back of the chair, like you do your coat. That's great.
There it is. So that's what we can find.
Now we're on this quite slopey here now, isn't it?
Just turn it around so you can see this slope. Get a better idea of how steep it is.
Cool. Should we just run along the slope for a while?
Yeah.
update for you, Eric.
Okay, thanks.
Thank you.
Amazing how different how it changed from when we're up on
Yeah.
the flat to now on the slope, where there's very little clinging on. If you can, you just sit down there wherever is comfortable there, and I'll have a look around some of these things.
That's good.
Or stabilise, as the case may be.
Okay, I believe the camera's all yours.
So there we
A little down.
I would like to have like actual lateral trip on. I know you're adjusting your control, but like actually a lateral, positive, negative lateral trip. Yeah. So it's nice.
We'll be able to control that live better.
Such a sweet little coral. Can you just take a picture and make It's very cute, little one. Oh, it's friendly.
There's an octopus now.
Other. Where's the octopus? Oh,
Straight in front of you.
oh, that octopus. Yeah,
Yeah.
cool. I'm sorry, I could only had eyes for the coral.
He's well blended in there.
He is very well blended in. Just in the bottom right-hand corner there, you can see a little octopus. Can you take a photo of the coral and then I'll focus on the octopus? Photograph the coral, yeah? Yeah, yeah. I can move to the octopus. Just chilling. Just chilling. Oh, no, he might have just noticed us. It's not a very big octopus.
No, it's
Where
not.
am I dot? I'll zoom out and get a scale. So yeah, he's about what, 5 centimeters across there,
Yeah. Not very.
if that. It's just a little one. It's a good spot. Well done, Russ.
Wonder what he's got behind him.
It's his little nest.
going to have a staring competition. It's going to stare at us and we're going to stare at you. Come on. Come out and say hello, little octopus. Let us see you in your full glory.
See, there are just some lights tonight.
There you go. That's perfect.
That's pretty reasonable. Take his head into his one.
Don't think it wants to come out and play somehow.
No.
Either.
He's
Suitably cautious about us.
He's really small.
right. It's back on.
Lots of good holes around here for things like octopus to live in.
that's probably.
Our most expensive piece of clothing is only $7.
Leather jacket, 300, but I've had it for 20 years.
Jacket wise, spent $300 on a jacket. One of the big police cars.
Yeah, they're good.
That'll last you forever.
That's that leather jacket, obviously it's 12 years. Good shoes like ever.
Yeah. Just have a nice little jacket. Wear.
So much there when you look at it closely, isn't there?
That's blue tag, that's the best stuff in the planet, right? Basically, you can touch that.
Okay, shall we continue along the
Certainly.
slope? Thank you.
Would you like to go down further, or would you like to remain at this depth?
You could go down a little bit,
Okay.
but yeah, just running along this edge for a little while would be interesting. So we're just having a nice afternoon exploring this eastern edge of Ashmore Reef. We completed 2 of our video transects this morning, and now we're just going back over some of the same ground, looking at things in a lot more detail, collecting a few specimens of interest, and generally just enjoying this moment. It's our second last day of diving here on Ashmore, so we're all starting to feel a little bit sad about having to leave soon and go home.
Is that?
It's holding that cruise very well.
Yeah. Should we cut the rock?
Stuck a little coastal arm.
Keeps the vehicle cooler if it didn't run on that.
Yeah, I noticed that. After I was watching too much drop. Just using the same amount of gas.
You're throwing it through twice as much because your pipe works. So as you're throwing your oil around, you're cooling.
Ooh. Let's look under that ledge right there. I suspect there's a cowrie hiding under there.
To judge. drop down a little section here.
There it is.
Got a shell in there.
That's what I was spotting.
Okay. Hold on. Just
That's a better angle anyway, actually.
there we go.
Oh, there's actually two. There's one there and there's one there.
Okay, I'm pretty stable there. It's kind of
Okay. Let's
like on my fingernails.
have a little look. What's that? That's a mollusk expert. Is that? No, that maybe not. That's what I spotted first time, but I don't think that's just a strange looking sponge.
Yeah, sponge or something.
Well, it's moving.
Here comes Corey near there. Corey.
Hello.
One of you.
Goodness, mollusk
Narrators
expert.
behind you? No.
Yeah, let's have it.
It's moved. It just moved.
Yeah,
Is that
And
a
there's-
crab?
I hope it's live. I'm not
And
sure.
there's another one there.
Let's have them both, please.
And I think there's actually another gastropod right there.
The one under there looks.
The one under there looks good. Okay.
But both,
Cody.
please. Yes.
I'm on it.
Your mission. We
No
should get some
pressure.
in situ
No
photos.
pressure, mate. No pressure.
Actually, if we can get rid of that swirly thing in the foreground
Yep.
first,
Yep, I'll go grab it right now.
then we can maybe get a decent in situ photo.
And that pink thing next to it's just a sponge, right?
That thing is just a sponge.
Just a sponge.
Just a sponge.
Just a sponge, and it's like
Just a sponge.
a sponge.
Just a sponge.
Oh yeah, and predictably there comes the thermocline.
Where the thermocline's from?
The arm.
The arm.
Yeah. It's the arm cooling in the water. Because it's the hot oil. Oil was like 63 degrees.
Beautiful.
Oh, now it's stuck to me.
Of course it is.
Yeah, I'm looking to move this out the way.
Alright, let's see if we can get a photo of this guy.
Little acorn.
It's moving like a hermit crab, but anyway.
It is, isn't it?
Doesn't want to focus at the moment. Is there any way we can it's just going to be bad with the shimmer anyway, isn't it? Yeah, it's definitely a hermit crab. I just saw an arm.
Yes.
There he is.
We might walk it out first.
Do you still want it even if it's a hermit crab?
If we can get it, it'll be good. Yeah.
Do suction
reason.
To the cup and then drop it in the. How big do you reckon that is?
It's a decent size. I might be able to grab
Yeah,
it.
it's not exactly tiny.
Should be
Hang on, I'll
fairly,
zoom out.
should be fairly robust.
Yeah. What about this small one right here where the laser is?
Yeah, yeah, there's that one there. So that guy's about 5 centimetres by the looks of it, that one at the back. Maybe we should
You want to take a shot of that one too?
grab the one at the front first.
Hmm.
'Cause that
Okay, because
guy's
he might be
good.
able to come out by then.
Doesn't necessarily look that alive either.
Is that a shell as well? For it?
Yeah, there's one right beside it, so a gastropod right there.
Oh yeah, another cone shell. Wow.
You want all those?
Yeah, that's
Why
okay.
not?
Yeah,
you want
that's
to take that as an in situ photo of both of them?
all
Thanks,
three?
Yeah, I
Gary.
think same quiver would be fine.
Okay. These will probably be going into quiver 19.
Quiver 19.
Awesome.
Now,
Right.
something that says it's already in quiver 19, but there's some confusion there.
Oh no, it's
was.
mixed, so there's a.
So there was one in 20.
Oh, okay. But I've got nothing written on this sheet for 20 either.
So 19 should be 20, just the lids were switched around.
Sponge plus maybe?
Yeah.
Okay, I'm going to go for the small guy.
This is going into 19.
Where did you think we were the 20 snail? Yeah, the 20 snail on the
It's
fan?
21.
Oh, okay.
Snail and Gorgonian went in 21 according to my records.
Putting on that there, huh?
It's okay. As long as you don't breathe. Approach out as far as I can. Rocking the camera back. Can you make it into there? Oh, whoa. Let's see.
I got him now. Okay, he is down here now.
Rocking the camera out.
Forge in a little bit. Keep coming. I'm at the porch all the way, actually. He's right on that edge. Look down here. There he is.
Oh. Close spot.
Better angle now.
That was intentional, right?
Oh yeah. 100%.
Apart from the rock.
The challenge with being stuck on a slope, everything drops down. It's gone.
Might have to suck that one out,
Yeah.
to be honest.
That one's probably going to have to be a suction. Let's see if I can
Yeah, that
catch
lump
it.
of rock's just in the wrong spot, isn't it? It's so close. Ooh, so very close.
Zoom in a little bit.
Nah, it's still there.
That's not him.
No, I could still see him sitting in this
Okay,
in the
yeah.
rubble there.
Can we suck up that one? Is that okay?
Sorry? Yep,
We can suck
yep,
him up,
sucking
okay.
up's fine.
I'll go ahead and grab move this out of my way.
Oops.
It's bugging.
Where did that hermit crab go? He skedaddled.
Okay, let's go ahead and grab this one.
always a sponge, isn't it?
Wait, is that the crab?
No.
Racking back.
Good porch.
Porch coming out. Nice to see. Okay, we'll stop the porch. See you right about it. Good job.
Beautiful.
Okay.
Next one.
So where did that crab go?
Where did he go? He's heading that way.
Sure it's not still in that area inside the underneath?
He was like right there.
Yeah.
And then
Can't have gone that
moving
far,
this
surely.
way. Oh. Porch him a little bit? Did he go down too?
Unless he's tucked in around that corner and we
I'm
can't
looking, I
see
don't see
him.
any sign of him.
I reckon he might be around the corner behind that sponge, because he was going that way.
Okay, well, let's take a look at
Oh yeah, arm cam. No,
I'm seeing him.
no.
Must be a burrow somewhere. You're not seeing him.
Wait, there it is. Yeah, right there. That's a sponge.
Wasn't to be, I guess. Unless he dropped down somewhere.
This. Oh, not there. Like, I wonder if he might have one down.
I would have thought we'd have noticed something that big drop down, but
He's done a runner, has he?
he's done a run.
He's fast.
No wonder.
We got the other little carrot. And
Yep.
we'll still work on getting that gastropods shell.
Alright,
I
let's get
think
some
a crab.
He might reappear, you never know.
True.
Jesus, go. Great herbs. Watch out, this potato. There.
Okay, push it.
Jeez. You ready?
Yeah. Okay, suction on.
It's all going into sauce.
5, yeah.
No, it's still there.
Still there.
Oh, it's got. Oh, I think you got it.
Touch.
Zoom in.
Yeah, I think you got it. Yeah, I think it's gone.
Alright, let's have a little look in the jar.
Look in the jar. Yeah.
Give it a little shake.
So challenging when they're stuck in little nooks and crannies like this.
Yep. The orange one.
Oh, yep, there. We can see him right there. Mission accomplished. Well done, Cody. Should we have one last scan for that sneaky little
Let's do
helipad?
it. Number 6 is open this.
Yeah, done.
Drop this.
Unless it's crawled up onto the roof, but I guess we'd have seen that with the can as well.
The down one is there. I don't see it.
Disappeared into thin air.
Nice ledge down here. I'm going to drop right down.
You want me to just go ahead and put a lid on that number 19?
I don't think that shell will float out in a hurry.
Okay.
But,
See if we fell down.
yeah, let's see if it didn't fall down. Nah, I think we'll give up. Give him a call
was
him
looking
a
for another one.
lost cause, hey?
I'm just looking for another
Oh,
one.
you're just looking for another one?
You want to watch out? Move it on. Put that
Yeah,
that
maybe
lid on.
put the lid on then.
Well, I guess he deserves to
Yeah.
Survive.
to keep going.
Yep, he tried that hard. He deserves to live. Oh, it's certainly a good spot for mollusks. Well, at least mollusk
Sorry,
shells.
moving out. Good job.
Okay.
That's too far.
Win some, you lose some, I guess.
Just get stuck.
Because you ate all the ones up around this corner.
These are open. These are open. I have this lid off, so it's ready to be sent.
I say you double dip, that was pretty cool.
The one time I didn't want to do that. We're heading down. There's a ship over here. We're just working our way through this like cavern. Yeah,
No, I'm gonna
is going
up
down.
and down and up and down. Yeah.
Got the ship station right there. Everything's going good. Thanks, man.
Yeah.
You're the man.
I've got nothing on except what I've had. Just been kind of go up and down, looking for interesting ledges and anything that might be. I'm looking considerably for shells. Shells right here. Lost them.
Got it.
She's been holding pretty good.
Afternoon, gentlemen.
Hello.
Just shift change time here at the mission control. We've now got Adam and Ben at the helm. Lots of little nooks and crannies around here. Those fish are in a hurry to go somewhere, aren't they? Screaming across the screen.
Are you happy to do that kind of up?
Yeah, no,
and
that's
down
pretty
the
cool
line?
actually.
Okay.
Yeah.
I'll do that then. Trying to give you that coverage there. Let me know if you see anything.
I will, don't you worry about that. Actually, can you want to just, if we can just stabilise here, looks like there's some interesting things just to have a closer look at.
Sure. Do you want me to land out and let you have a look around?
Yeah, sure, that's good if that's easy enough.
Sure, can do that.
Just settling down here so we can have a little bit of a closer look at all these wonderful nooks and crannies.
Sorry.
That's all right.
2 seconds, I'll just land you back.
Challenge of trying to hold an ROV stable on a vertical wall.
I'll push your camera up to you. Nice close picture.
That's good. Thank you. These little arms waving.
Bridge, ROV, that's an updated waypoint.
Upside down the crown.
Thanks, sir.
It's amazing how big the tiny corals are. You know, again, used to see bigger ones because they're on the chicken. That's a cute little fish. Yeah, not bad for a fish. They drink. You've heard it here first, folks. Narrator Wilson just called the fish cute. We did see a really cute octopus. It was already in its lair. It was sitting beside a coral. It was my we noticed. Well, I didn't. I saw the coral. I saw it. Just sits there. Is it grabbing little bits of plankton? What is it just doing? Either floating with us or trying to scare us away. I've only stopped twice on the wall. I sort of went along for a while and I just got down a bit. The first stop there was two cowries and a gastropod. Except one of the cowries was a democrat and ran away. look for, but I just noticed that right beside it was a very tiny little etching test. Suck that up then. I think we'd like to suck that up. I suspect this might come with it.
Perfect. Yeah.
For once we've picked something well positioned.
There's already one.
You want to suck it, you said?
I think that's going to be the easiest. We are, or it could go in with 5, which is just a back of the chart. Okay.
Yeah, it's on jar 6 now, but I can index the 5 if you want to put it in there.
If we can tell apart, sure.
Pretty different.
Okay.
Even I could tell them apart.
Jar 5 would be great.
We didn't get a photo of it. Did you get a photo of that in situ? Or shall we?
Sorry, we didn't know whether you were listening.
Yeah, sorry. We weren't very communicative about that one. We'll just, once we get back on round talk, we'll take a photo first. Photo taken?
Yeah.
Lovely, thank you.
Ready to roll? Okay, jar 5.
Yeah, jar 5.
And is it the shell or the urchin you want both?
It's the shell.
The shell.
It's the egg or the shell?
The urchin, shell. The shell.
The pink thing, not the white thing. Pinkish.
Ready? Pull on.
Just at that wrong angle again.
Here, try to, I'll just wrap it a little bit, try to give you a pitch underneath. What's that? That helps you now. Ready? Go on.
Are we winning? Nah.
We will.
I know how determined you guys are. Ooh, that looks like it got sucked up.
See it? Just give it a little shake there. I'll just make sure. That's off. We'll just have a look here. Let's see what we're saying. Still on? I think it's in there. It's up to someone. No, it's off. Now it's on.
Which jar are we?
It went into jar 5 with the other shell. In theory, we'll just check it did land in there.
Okay, there it is. Is that it? Right, keep indexing it. We'll see if we'll get it.
Very hard to tell. Looks like the shiny one in the back.
Yeah, I suspect it's in there.
Oh, sorry. Too close. Too excited.
Turn it on again. Turn it on. Yeah, it has to index it out. But I can. Yeah, it's not right. Did we get that carrot? Catapults. I suspect it's in there, it's just in the
Yeah,
bubble.
it's probably just buried. There's 2 shells in there. I can't see either of them anymore. But
5 to 6. Okay.
cool, thank you. What sort of shell was that?
I think it's a
It's still there. Okay. Finished. Alrighty. Shall we continue along the wall for a little bit?
Oh, I'm just watching somebody's
Sorry.
just
Oh, okay. There's a couple of sea stars that are marked always away.
Did you want to see the substance?
That's quite good.
Yeah. It was double sound.
Before we got to this one,
I have photos of them if you want to see this.
because, yeah, let's just keep meandering on the wall for a minute. Oh, can we just stop and put what that is? You can just quickly zoom on it.
That's all you.
It was a shell once, but it's broken now. Okay, thank you.
See them? Should be up there. There's 186 of them. They're not up there.
Okay, so yeah, I've just been heading on this wall, but I was planning on heading back up the shell. Yeah, I think, oh, absolutely. But yeah, once we've wandered along here, I can just pop up above the transect over about 80 metres or something and see what's up there. I usually manage to find if you get a chance. It's so fair. Maybe that's no problem. That might be something to do.
that this week because I just came up here a bunch of meters, so I'll drop down and just see the total meters. Try to keep it like I'm thinking about 12, 15 meters. Try to keep it like 65 or something. So it does. Keep one hand on each keyboard. That's not yes.
Need a break. Need a break.
Yeah. Where did you see it? Rams coming up.
It's been a very nudibranchi morning.
What's in center screen there? Is that the neighbor?
Yeah. Is it a familiar?
Oh, yeah.
Oh, your cute fish there too.
Someone cut it.
Got it in situ, shot it back.
Yeah. Curled underneath.
No, I think somebody cut it. Had a piece of
I think you probably such a sound there.
Awesome. Thanks, guys.
Do we know what genus this one is?
Kind of looks like a retrieval.
that's the James.
Yeah. Yeah. Could
I
be.
asked to write it, but also because I would like to know study.
Awesome.
Do you want it?
Yeah, looks great. I think the chances of being able to collect the sponge as well are very low. So I would probably suction, I think, with that guy. If I'm not mistaken with the size, which has happened before.
Sure. Do you want to double it up on one of the other jars?
Maybe not. Maybe 6.
Yeah, just pinch it basically. So, sorry, did you want to double up on a jar?
No, I'll probably keep it
So we'll go with,
in 6. So
Sure.
move to 6.
Yeah, I'm on 6 now.
Oh, cool. Perfect. Thank you. Yeah, don't think we've had this guy before. Oh! Sorry. I didn't mean to scream out. That was completely involuntary, people. Thank you. That's
And
great.
you did. I saw.
Awesome.
Nervous.
I'm sorry.
Okay, indexing to 8.
Just gonna zoom in
Yeah,
so we can
first.
see a feeding star. It's probably not though, it's hard with these ones. It's sometimes quite hard to see if these suctorial feeders have been eating into a sponge. It's a lot easier to tell.
Straight up.
Okay. Hello, snake.
Snake button. Snake button. Snake button.
Oh wow.
Nice. That's an older snake. Pretty.
I think we should capture it. Oh.
I've certainly come to appreciate olive sea snakes while seeing them from an ROV. I don't appreciate them in the water when they're looking at it.
I would concur with that.
Yeah, but maybe next time I'll have a... Yeah. I'm getting better and better with them.
Have you ever swam near them?
Yeah, yeah.
Very curious.
They're very curious, which is what disturbs me most.
Are they curious and deadly?
Look, I think an underwater interaction is so unlikely to result in any injury, more to the snake by you panicking. But they are venomous, and from time to time people are injured or pass away from mainly pulling them in nets and getting an accidental bite.
Right.
It's just gorgeous to watch them forage though. Just move so beautifully. Wow.
Just want you to know that we've caught eels before.
We don't have any permits for vertebrates, so we'll definitely let him on his merry way. Ooh, scratching, having a little scratchy scratch. Oh, that's
Now,
so
does he
pretty.
have the surface at all?
To breathe, yeah.
So
Yep.
that's quite the depth.
Yeah, it sure is, 106 metres. My understanding is that, and I'm sure it might be a little bit different for different species, but they can hold their breath for up to 3 hours, but if they're actively swimming, it's more likely 30 minutes.
Right.
Yeah.
Did they have any predators?
Yes, they are fed upon by sharks, actually. And people.
People, is that? Yeah. So you
Cute. I just love these sponge forests.
do you want to bump up and take a closer look at them?
I think it just looks like an amazing scene.
So now that you said people net it up, have you tried it?
No. Nope. Seems like you could shoot a shot of that. It's so gorgeous. What an amazing sponge garden. Absolutely gorgeous. It's like a little fairy wonderland. Hard to imagine. Here it is.
That fell on the floor, that's what I was saying. I was wondering where did that come from? Sounds good. Oh, weird.
I can pull the rails in and give you a closer look if you want to look at that sand.
Sometimes things are best left a mystery. I see the shimmer is strong today.
Shimmer is strong with this one, yes.
Oh, wow, that's so cool. Thank you.
Back to our transit.
Well, maybe along a tiny bit more. It's a pretty cool spot.
Yeah, just been going kind of all the way up, over a couple meters and all the way down. So
Yeah.
just giving the whole.
Sounds awesome, thank you. Sounds like my idea of a scenic drive. Wouldn't mind having a look at a couple of fans, so if that's okay to start, that'd be great. Yeah, I put it together.
Take it off photo. Let me take it off photo.
Sorry, what?
I'm gonna take it off photo and you can try it again.
Oh,
Okay, try
it's
that.
working for me though with manual and auto and yeah. It's not the same as the gastropod that you guys saw earlier today in a
Oh, nice. Strange guys up there. Why don't you guys on sheets? It's alive.
I don't know.
Probably worth getting. It's up there, it's probably alive. Oh, you mean the one on the microscope?
No.
Looks to be a little bit alive.
Well, it could be a Hermie.
Oh, okay.
But yeah, just take a front. I'm not sure yet. Kind of looks like there's another one back over here, Corey. Maybe.
Photo of that one.
Maybe not.
Sea snake.
Oh.
Right here on the rail. Sorry. Snake.
Oh.
Have a That's that one.
We find out later. It's the same. He's so cute.
Do you want to try to turn the ISO down a bit? Are you happy with that?
Oh, I'm happy, thanks. Yeah.
It's definitely hanging out. Notice how he can like become buoyant and then instantly
boom.
heavy.
Yeah, yeah. The drop
Like, how do
to the
people do
ground
that?
is amazing.
And it's like it's not even that he's swimming. Sometimes he just floats up and he goes.
Yeah. I'm not quite sure how their locomotion works. Yes, I would really love confirmation about how sea snakes shed their skin. If anyone knows, I've been trying to find out. please. That looks cool.
Want to take a look?
Sure, please.
Sorry, I stole this.
I was wondering what it was. Thank you. Wait.
No more?
I don't think it's Russ's.
Somebody's head perk up over there?
Usually when he's doing the arms, I'm like, wait, he has to do sampling.
Whoa.
Don't worry, Ben will eat it.
I'll find it before.
Thank you.
Stole the stash.
From J-Ron, thank you.
They're all the good colors, thanks to you.
That should show up. There's one green one sitting here with an empty package. That's just like.
You've got a snappy time of day here. Oh, look at this little. It's also coffee time.
Keep looking.
He just looks so pleased.
Sure, it's pretty classic.
Can only do one a day. Oh dear, the smell of coffee is lovely. See if we can smell an ovule in somewhere.
Do you prefer up high or a little bit low? Been going all the way to 10 meters and kind of doing the wave.
I quite like this actually. I'm happy to linger.
I'll stick up here. Maybe just a touch northwest. Much nice and slow on top of the ridge.
Bridge, I just updated your position.
You'll actually be closer than the
So I'm a little bit interested in this lump on this one.
Okay.
It's a bit hard. I don't know if it's possible to come completely almost like 180 and face that.
It's going to be downhill in that direction, so it's not the greatest way to set up because we'll be sitting like this.
Oh yeah.
Yes.
So how do you think the best way to get a good look at this is? Because I think if we come that way, it'll be kind of hidden behind that.
So show us where you're trying to look at.
Just get it there eventually.
You talking about that small animal, the white one?
No. So it looks like this is a bivalve sitting on the fan there, but it also looks like there's something on the other side of that stem. You can just see it poking there.
Looks like it's stuck on the other side of it into the coral.
So it's not like it's not a thing like that. It's a thing like that and a thing like that.
Okay. I can bump over a little bit to the left there.
Yeah, because we might just with a different view we might be able to figure it out.
Sure.
Did we want to in situ sort of that?
No, I can't see anything yet. That's a problem.
grade is it?
Nothing big.
Do you want us to take a snip of it and hold it in front of the camera?
Is it 2? Is it?
I think it might be 2, yeah.
Technically, ice.
We may leave them there.
Do you want me to try to get a little bit different of a view? I can slide
No.
around to the right side.
I think we've figured out what they are.
Okay.
Yeah. No.
Moving along then?
Yeah.
Alright.
It's quite hard. I mean, every sample is useful, but we have to make some tough decisions about what mysteries to solve and what to just move on to look at new things.
There? Which one? The little white one? Yeah. That's right.
You might just be able to see with the flies through them. Yeah, nothing.
Okay.
I retract that inquiry. mean, by the time we find the market, it's like that.
Yeah, true.
But it's situ that.
Thank you. Oh, there's a nudie.
Yeah, that's funny.
So I guess the question is, are they both in reach with the suction?
Yeah, where was it again?
I think we can pick that up.
Yeah.
That rock. He's on the rock.
I think it's a bryozoan, which is pretty fragile. But could work.
Put him in quicker.
Yeah, okay.
Okay.
Cool.
One of those dark chocolate truffles. Didn't get enough. I'm gonna pop over to the left a little bit because I can't approach your anus.
You do that.
Punch it. That's right. Okay. You see it?
Yeah, I know where it is. You might need to move forward a little bit.
Where is it?
Like 7.276.
Okay. Oh yeah. Okay. Okay, coming up.
I guess.
Should be good.
Okay. First, you can try to pick him.
I lost him there. Oh, I see.
What's going out? Or do you want a second?
I think he's got 17 so.
Okay. Volume.
always bigger than the animal. Yeah, I don't know if I could pick him up. I thought he was smaller than that.
Surfing?
Yeah, I was surfing.
Okay, torch coming out. Okay, so we're on jar 7 right now.
Okay, cool.
And how do you feel about picking up or suctioning that other shell or something?
Circle shape.
Off and on a bit.
I didn't see him go in.
It's not come in.
Look at this camera here.
Oh. Is that? Oh no, not good. Oh, there we go. Yep. There he goes.
Shoo!
Awesome work.
Okay, so I will index to jar 8. Which is our last open jar, I believe. 8.
How did you want to collect that other snail shell? One of the first.
Oh, that one. Oh, okay. You do want that one. That one we can. What do you think, Ben?
You might be not in the right spot for it anymore.
I have to bump down to it, but do you want to scoop it down for something? It's down there. I have to go back. Okay. Other shell.
There's a sponge right inside.
Yeah. Okay. Sponge. There.
Oh yeah, yeah.
Okay, I am Steve. Do you want to just slip them in the same jar?
Can
you
do.
can tell?
Yeah.
Okay. That's easy. Just do a half step there.
Jar 8. Jar 8.
I'm sure it's in there.
It hasn't picked up in yet, maybe.
Oh, okay, just check.
Just bring it around here.
No, no. Try the old off on.
There he is.
Yep, there we go.
Confirm jar 8.
Yep.
Beautiful.
What was it?
Had nice bit of coloration in the imagery.
Oh, okay. Freshly dead. Maybe live.
Yeah, maybe.
Okay.
Move it along?
Yeah, thank you. And Adam, I know Karen was keen to come up into a shallower region, so I would probably could just creep ahead if that's at this speed, of course.
Do you want to do the west? Maybe. Do you want to do west over the ridge yet? That's a stream. That's just going to see the arms out there. Check the camera. Yeah, we're going to go over on top. You're in straight west, very close. I know the straight west. I'll kind of meander from one place, or sorry, back and forth west. Yep. Or even a little bit further west too. 101 way.
Ridge, you have a new position?
Keep moving the camera on to the right. Yeah, you did great. That's the final shot. Yeah, it came to check it, didn't it? It's a nice area. Since we're on top, we're not putting more data around. So we're crabbing sideways over the top here.
We're gonna head west now.
Heading west. I walk down the, back over, shallower.
Yeah.
Okay.
So, are you? Is there a thing happening?
No.
Is there something? Oh, okay.
We're just taking it when we start moving. I just like to make sure the arms out of the way, but they've already done that.
Cool.
Would if we get to the point where we're not seeing much new, then yeah, of course I'll take a chance and do a quick mowing of the land somewhere nice. If we see something particularly nice, or just let's see what we're going to see, something to sample.
What time are you guys doing the CTD at?
Oh, are we doing one today?
Yes. As far as are we? That's what was said in the meeting today, but maybe confirm that.
Okay, that was not passed on.
There's one minute to leave right there. Let's find out.
What does that mean we have to move off to do that because we're not in the right depth? I'll ask her.
Just thought I'd ask in case we get too far up from the
Yeah.
shelves. We could just
Very good
zip down
question.
there.
Thank you.
You're welcome.
Okay, so Karen said yes, we do need to do a CTD.
Okay.
So, I don't know, do you have any information about where the ship needs to be? Like, do we have to move off?
Do you want to go and clarify that CTD information? To go check.
With who?
I don't know, Karen or...
Karen has said yes, we are, and so now I'm just trying to figure out how.
All the previous ones, we did 150 metre water depth.
Okay, it's Corey here from the WA Museum. We're just gliding along looking for some more invertebrates. Just some discussion about the location of the next afternoon taking place.
Oh yeah, yeah. More important is
It's been a relatively successful day so far. Quite a number of nudibranchs have been collected, and assorted gastropod sea snails as well. So it's quite a busy bottom that we're looking at here.
Yeah, I'll take it. It'll be different than what we're actually absolutely. Alright, that's just going straight back over this line.
We're not sure if that last gastropod that we collected was live, so hopefully we'll have a closer look when we remove the material when the ROV hits the deck later on this afternoon. And
There's been a mutiny.
and I will just put you back to narrator.
Well, we haven't got off the top of the cliff. Are we still hanging off, hanging off the top of the cliff?
Do you want me to go back and forth on this track line?
I'm waiting for
Okay.
instruction.
Yes, so we have a plan. We still need to do our C to D at 150. So instead of kind of slowly creeping down and out, I think we're going to do a bit of a fast blue water fly out to get into position. We can drop down then or whatever you want to do, but then we'll do the C to D and then come sort of fly back into this top part of the cliff as soon as possible.
How about we do this? We fly out like that, and I'll go to the bottom, and we can do his calibration while we do CT.
Oh yeah.
Okay. Alright. I'll see you in a bit.
Sounds like a plan.
Thank you very much.
Could be cup of tea time, depending on your tastes.
Alright.
Bye, Nudibranchs. We'll be back.
Thank you very much.
So we'll, yeah, if you follow my screen, let's zoom out.
Head east,
Yeah, if you just bring him straight east, and you don't even have to go that far. Does it have to be exactly 150 or can you do 100?
Oh, if we get you guys, the vessel is at 150. That's fine.
Yeah, it's back up to that line. Alright.
But
Oh, snake, snake, snake! Oh, I think it's the one with the funny face. Let's see if we can get a look at him. Ah, don't go under us. Ah. Oh my gosh, why does that happen?
Snake it on you.
I just get overexcited, I think. Start twirling everything. I think you just do it ASAP. Except now we're engaged with a snake.
Bridge control. Yeah, Eric, I think we're gonna do a CTD, so we have to travel back 400 meters directly east to get to 150 meters. So we're just gonna fly as fast as we can and just check this out. You can move the vessel as fast as you can for the sake of time.
It's much over, so it won't be very fast.
Roger that.
But yeah, so I understand that the CTD will be done immediately after
As soon as we get off, I already moved it east. As soon as we get to 150 meters.
and if you guys go out quick, due east from here is the shortest point to get to it.
Yep.
Okay.
Where's that? Straight east from us?
Yes.
Yeah.
You ready for the marker over there? So, okay.
just go down the streets. It's gonna drop off here. This the next slump is 20 meters. Found a little. How far out from our position is 150 about 400? Oh, it's that far. Yeah. Meters every 2.5 minutes. Yeah.
Enjoy your time off. Good afternoon, scientists.
Good afternoon, Cody. How are you?
I'm good.
That's good.
Settle down, children. Keeps touching me. Keeps trying to feel my muscles.
The line.
Do you want me to put masking tape down? Okay, just had a question come in about the water temperature. Right now, it is 23 degrees Celsius. Very rowdy. Oh, yeah. Thanks, Adam.
We're just going to go. It won't crash.
We're going to see the
Welcome, welcome. Team Texas. Back
Hola.
again.
I have a picture of me and you feeding each other from
No, we're handing each other lollipops. So where we go?
Just going out to do a CTD.
Where's that at? Which direction?
150. Over to
It's not a matter of where it is, it's a matter of how deep it is.
Yeah.
Oh,
Alright, it looks like you got a bunch of nudies.
I have. It has been a spectacularly great gastropod day.
Yeah, we got a few of those shells.
We got one of my favourites from this morning was a eulimid that was one of those parasitic snails that's perched on the top of an urchin, just sitting right in the middle like
Just
this little king of the castle. Yeah.
Slippery's version.
Very cool.
That's awesome.
And there's some suspicious white extra bits in the spines around the side that could be other parasites.
you look snaky to me.
Where'd you go? Where'd you go?
No.
Not. Did you find another snake?
I found a not a snake.
Hey, snake over you.
You got a lollipop?
Snakeometer. Why are you?
It'd be so nice and bring you one.
Oh, strawberries and cream. I didn't see those in there.
Would you like a strawberries and cream?
No, thank you.
Strawberry.
I've just had.
Take my lollipop back.
Don't want it to go to waste.
There's evidence of other snacks.
I just want that camera for Ben if you want it back. That's cool. What the heck is
Did you get my photo?
Yeah, I did. That's hilarious. He made that figure to put on his food.
Yeah, it must have.
And so a follow-up there for Elizabeth Harrison. All of the CTDs that we've done on this expedition have been down to 150 metres, and that's to correlate data with the transects that we're doing. So we haven't done anything deeper on this trip.
You never saw those?
Oh.
Sticking at your booty, dude.
Get paid.
Okay.
No. Probably get paid one day. Just about to put in my third invoice. Fourth invoice, actually.
So that waypoint that you started just 150?
I just moved it. Just looking for 150 more.
Yeah, if they go too west or too east, they'll get to it. That's what I gotta do.
You're thinking?
I wanna go gully there.
Give me
by Sea Star Power.
Sea Star with power.
Ben
By
already
the
made
power
one,
of Sea
so-
Star.
Should I just move his final waypoint then? Bridge RV.
Go ahead, Cody.
Hey Jason. So it looks like we're going into deeper water, 150 meters, to throw the CTD over. I'm just going to move your final resting waypoint here, roughly. Can you see it? Yeah. Yeah, roughly. It looks like it's about 260 meters or so. And then we'll just keep up with you. I will get back to you on that, buddy. You're welcome.
So planning on
Are we still planning on coming up at 1700?
That was a head nod, yes. Head nod, yes. Over. Send us some lollipops. Speaking of lollipop lamb tonight.
What?
Lollipop of lamb tonight.
Oh, I know.
Fish.
Chicken.
Does that mean? Cases.
I hope not.
I'm going to pretend like I didn't.
He said Darwin's on lockdown. Possibly.
Who? Who's?
Oh. 13 cases.
13 cases of Darwin.
But they're not quick to
Okay. I divert to jump.
Have you done?
Yeah. Yeah.
You like the little sponge cup?
I love it. I can think of all kinds of experiments to do with it.
Johnny from
Urchin, dead.
office.
Didn't hear you just say.
Yeah, go.
I don't remember.
There's a couple. It's gonna kill me. The boat was inside, and that was the bar.
Oh, the hula hut thing?
Just an FYI.
The rum bar?
No. The one, which one? Old girl tennis table.
Oh,
Rodney's
yeah.
favorite.
Rodney's.
Right,
Yeah.
yeah. I like Rodney's.
Yeah, my mate's right. It's really good. Good vibes, loud music.
Taylor took us there.
Taylor took it. That's right. I do. They had the fish raffle.
We did not win.
We lost horribly.
How good are that chips? They're like triple fly fried.
I had no idea.
They had food?
Yeah.
Oh. Oh, yeah, they shoved food down our throats at one point.
Might have not.
It's gonna be another 150, but it's what it was.
bullshit function comes in.
There's a button.
They're on altitude clock.
What's that?
On altitude clock.
And cruise. So once I figured out where I like the skin. Steady as she goes.
So what do we? We're at.
Steady as she goes.
Oh dear God. Sing along. If the sing along gets too hard, people at home, I'll just turn the mic off. We are currently cruising at an altitude of 1.2 meters, moving at 0.2 knots. No, 0.2 meters a second. So, hence the relentless sandscape that you are watching right now. We're still just moving into position to do our CTD. So we are just steadily moving out there, trying not to look or see anything.
Steady as she goes.
Steady as she goes.
Is this camera working good? What was that last
It
time?
is warm in here.
I think wasn't it something bad?
I remember it's like unplugging that Ethernet cord.
But all cleaning is meant to be the 29th.
Gotcha.
But
Yeah, no, I can drive close to it.
Nautilus shell?
looks like it. Nautilus.
Yeah, cool. So why do some sink and why do some float?
What's that?
The nautilus shells. Why do we see some on the bottom and some float on the surface and wash up on beaches?
Levels of decomposition?
They have like chambers of,
What if it gets cracked?
yeah.
Floods?
Or is it liquid? I'm not sure.
Somebody in the community knows.
Well, yeah, it definitely has an air chamber because that's what it uses to go up and down.
Hmm.
Air chamber gets flooded. Probably like submarine.
Kind of exactly like submarine.
Just a little bit.
There was a crab staring at us.
They find the Indonesian submarine?
What?
The Indonesian submarine that's lost?
Oh, yeah, that's it. That looked like a sea cucumber. Way out yonder. Are we looking for anything while we do this transit? Or no?
We're not looking at anything.
No, so if we see
Once
anything.
we stop. Yeah. We're just trying to get the CTD done as soon as possible, and if sort of, I suspect at this point in time we won't make it back to the wall after the CTD. So that might be a bit, I'm not sure.
How many pets do you have?
Oh, enough.
Have you,
Pretty
named, have you named them?
That's what I do for a living, J-Ro.
Well, let's see.
But it's faster, it's a bit fast. Usually it takes much longer to name them properly.
No, I mean fun names.
I don't do fun names for study animals.
The
It
starfish
seems a bad
is-
idea.
The sea star is Raffy, right?
Well, I made an exception for that one, okay.
You can call him Spot. Oh, looks like a spot.
That's Shelly.
Snow
Shelley.
globe.
That's a snow globe, yeah.
Shelly Michelski.
Oh,
sponge.
yeah.
That was, yeah. You did that, didn't you?
I did.
You did that.
The sponge.
What are all these
Oh,
little
no,
guys?
take it. Look at them all. So
How
many.
long did it take?
They like the light.
Life if the borders would go. It's a 2-day journey from here.
Look at this guy.
Oh. So more, way more. But also, you get to the border, you won't be allowed in. So.
Yeah, you know, so
yeah.
it was just if the borders would go, how long that journey would be, is it?
Oh.
Spend a few days in transit.
Depends how crazy you want to go.
No, I don't want to talk to you either.
I would say minimum this, probably. And I think at this time of year, it will rain happen.
Back on 8. Camera goes out at almost 0.2 meters a second.
Oh, 4 rock lobsters.
Almost seems like you're staying still.
Star.
6 more meters to go.
Philadelphia,
Depth-wise.
Yeah, it's quite a gradual slope here. So that's that white line is the 50-meter contour, or 150, sorry. Here, that's the all top part of the sun. It's working out planet size.
Diadema!
Bits of plasma.
He's got 2 little eyes looking at you.
Right.
Hi!
Could potentially use large flares.
Polar bar. See how we're going back to the moon? See how we're going back to the moon?
Yeah, there's 600 or 6 guys.
2020-ish. Once he's done with the space, it's a Mars city or whatever it's called, space sequence.
Jerry, you,
What are we doing?
octopus.
No? Hold my breath.
Beautiful.
$2.
All $2.
Cool.
This little chair.
Borrowing an enemy then.
Hemis.
Hi.
Give him from it. Hemis. It's got a alien on it. It's good.
Nice. What a nice little home. Cool. It's like hyperventilating looking at it.
closed his eyes, he's not like the bright lights. Or she, is that a brood? She's keeping cover in.
Maybe.
Nice.
Great thing back there?
The shells.
Behind the shell. Great thing.
It's got
Really soft kind of flat. Might be.
Yeah.
Might be what?
That looks a little back on the right there. Oh no, that might be their arm.
That's
It's just the arm.
Oh, back there.
Oh, that. It's hard to tell. It looks like suckers.
They only have one exit for the aspirating. Aspirating.
Thank you, sir.
Hanging in. You get a little carried away. Urchin.
Purple urchin. He's moving fast.
He wasn't moving fast. Sorry, John.
Almost there.
Great.
40 meters.
Oh, the zigzag lines over there. Soft to the side. Looks like it. Maybe it's just the way the line is.
So that means we're going to be diving for 6 hours. Give it to them. And they dive for 6. And then what comes up?
Or they just want to dive whenever. They have to be ready to go at all times. I don't know. It's never actually been 24 hours.
So that was a 24-hour cruise too. Randy did it. I think that's what it was. We were diving 40 different times. Like, fortunately, we'll do it with her. You'll grab the big screen tonight. Throw it into the
Right. That's right. Then, I think you might want to set up that. Anywhere in particular you want to go look when we get here?
Oh, turn left at the big sand grain.
You want to what?
I said turn left at the big sand grain.
Night Star Power.
Really? I mean, we can just sit down and maybe we can use the jaws to like sweep the sand, see if we knock up any bivalves. We can do useful things.
Do we have like a little fly around in the area once we get
Yeah,
there?
sure. With that mysterious meal. Yes. Oh my god, could you imagine? If we stumbled across a meal right now, I'd be very excited.
It's in public.
Yeah, let's just fly around until we find a meal.
Look at this horse. I think the cups might shrunk a little.
I'm sure.
No.
The small one's good. Chris, we're still I would.
Snooze, you lose.
Now that that
Are you sure? That looks like something weird.
It's nautilus shell, but sort of broken, I think.
Oh, okay, I see it now.
Yeah, it's like the underside, so the top part's been broken away.
Good.
But yeah, pretty weird. Oh, what's that shell there? There are actually quite a lot of shells.
Oh. Is that a shell right below it?
Yeah, that's what I'm looking at.
Yeah. Bluish.
No, but it's interesting.
ship is setting up.
Have a zoom.
Are you guys gonna stay out for the winter now?
Yeah, you're fine.
Oh yeah.
Actually, let me
That
just
is
drop
interesting.
No, you can change that. We have our attention over here.
Yeah, for the recording of it. Just in case.
And I'm gonna call that good pick down.
I do show one of the 2-inch letters.
Debris.
Yeah. We definitely want to collect that little shell in front of the Nautilus. Oh.
I saw some of those earlier too. If you want to go see more of things that look like that, like
That is interesting.
river stones of the ocean.
What's
Is
next
it
to
a
it?
coral? It's a little coral. Oh, it might be. I think so. It's striated. It's just got that little yeah, it does.
Boom.
Well, if you're going to bring some sour worms, I'll donate some neither. Maybe I need
Well, nothing but.
and there's nothing but
Do you want to check the bridge, see if they're set up when they want? Here's the thing, yeah.
Bridge, RV.
Yes, go ahead.
John would like to know if y'all are set up where you would like to be with this.
Yeah, we're good to go.
Roger that.
John.
Yeah.
He's good?
Good to go.
Thanks.
Ghoster.
Yeah, so.
Okay, we would love to suction up that little shell.
Really don't.
How would you like to get them suction or?
I think we better suction. So we could still use 8.
There.
And then we've got a thing we'd like to boop.
Think about what?
We've got a boopable object after that.
I'm thinking about the door. You gotta bring it. When you get to bring in. That's 2. 2 for the red contours.
Tell me when to set.
Liz, John. Hey Liz, the bridge said they're set up, ready to go whenever
The cloud went in, so did the shell. Capture.
Thank you.
Got a half index. Half
Oh my
index.
gosh, look at the time. Okay.
We can
yeah, whenever you guys
leave
are
it
ready,
on
we're
8.
station
That's a
now.
coarse one, isn't
Okay,
it?
and set up and stuff.
Yep.
And there's just dead shells in it, so
Okay.
no one will get hurt.
Sounds good to me.
Awesome. Alright, now boopable object. We might not be close enough, but this.
Is that John too?
Oops.
Gotta do some booping.
Yeah. Can't tell if it's 5 or
You know, jaws kind of look like a crocodile or an
They
alligator.
do.
It is. It's a little crocodile face. That's all we see. Crocodile.
That's what you see too, right? Yeah.
Oh yeah, everybody sees. No.
I thought it was just me. I felt special, Karen.
Especially since his head is red.
Yeah.
polish on. Yeah.
I felt special. I thought it was just me.
It's a crop
Cody,
lipstick.
you are special.
Thank you. What I like to hear.
But hang on, let's just have a look.
No boop. Boop.
Wait,
Boop.
Oh, that's coral. That's coral. We don't need to boop.
Okay. Yeah, now we've got that angle on it.
Coral confirmed.
Yes.
No.
Sorry, no
It's just
boop,
the
no
straight-
boop
it was just the
situation.
straight
Is that
angle
flow?
track, isn't it?
Is that flow? Flow Bella?
Yes.
Blue bellum.
Well, I don't know. Ask the coral person, not me.
Can it save you money on
The
your
belly
insurance?
day. Compress that
Oh,
way.
good there.
Yes.
Progressive. Here.
Oh, they don't have progressive over here? They don't have.
Find a place of nothing.
Would you like to see whatever that
Operator.
sea urchin shallows up there?
First, I'm just going to have a teeny look at this sea star.
Powering on CTD.
All the buckets in the lab are filled with the warmish water, so just need to cold
It's quiet.
Thank you.
Went from being really loud to being
You
quiet.
know, last night.
Oh, you're going over to it. I got to stop touching the camera.
Really, really quiet.
I'll get you. If you want to see it, I'll take you to it. You're there.
Oh, thanks.
I came out of my daughter's there. And of course the
Why am I doing this? No, also the wrong way. No, end of day is very hard for motor control. What is on its side? A beautiful big scale worm. My God. I think that is a big scale worm. It is beautiful and I must have it.
Is that maximum effort?
Yes.
No.
Oh.
There was a little bit more.
There was a bit more to go. Look at that.
Do you want the whole thing?
Oh, yeah. Absolutely.
Is it quiver size?
It's pretty small.
That is so cool.
I'm just trying to get to
Stop moving so fast.
these things.
Stop moving so fast.
Oh yeah, that's gonna send a quiver.
Yeah.
Is it quicker?
That is so
Can I go for a grab?
Can you grab it?
Yeah, yep.
Let's go.
Nah, he'll stick.
Just ducked around further down.
Yeah, he's feeling for it. He'll disappear.
I'll copy CTD and water. I have to go down to 10 meters and hold it. Just
There he is crawling around the other side.
Yeah. Oh my Lord, look at that. Wow.
Oh, it's huge.
It's beautiful. Ah, beautiful. Sorry.
Uh-huh.
For sure.
Zoom it over, make sure that one's still on.
osmosis go through it over.
He shoots.
He scores. Thank you. So, let's have some tea. Well,
Exciting.
for a place with nothing, it's absolutely high excitement. Must be the day we find the Mueller, right? Last chance, Larry.
That translates for us. Yeah. Or we could recreate it.
Great work.
We're still in the bio-box here. What are you doing?
I know. Just got, well, this happened because I was mostly in the lab this morning.
You're just saving it up for that meal, aren't you?
Good to come up. Oh,
Oh, yeah. I was like,
Good.
I don't think so. Yes,
Another
let's
20 minutes, yeah.
let's find a Milo. Let's do it.
Find a what?
A Milo. This
Operator
big
control,
volute that I'm
you come back
interested in.
to
What does it look like?
Surface and
It's
zero
like a
wire.
watermelon. It's
Okay.
big.
Looks like a watermelon.
Yeah, we'll see it. It might be smaller, slightly smaller than a watermelon, but it's a big volute.
Take a taste of that watermelon or
Yeah, right. Is it green or red? Or both?
The shell is sort of orangey and the foot is often black. But no one in the live animal, or there's no images of the live animal, so yeah.
Okay, it's fine.
Couldn't say for sure.
Greater.
Maybe it's in there.
Yes, people of the chat line, I'll have to agree. I've been really impressed by the diversity of sea stars here at Ashmore Reef. I would love to spend a bit more time collecting them and imaging them properly, but as usual, trying to do everything. So hopefully you've seen a smattering of beautiful stars, particularly like some of the Nardoa that we saw, I think, just yesterday. Yeah, super diverse area for those, and likely that's due to sort of the influence of sort of that high Indo-West Pacific proximity to Indonesia, just big diversity in echinoderms. See, there's a good track. What made that track? Maybe it was a meal.
There's tracks all over the place.
Yeah, they're probably sea urchin tracks, but nonetheless,
I'm on my way.
Big thing.
just in case.
Oh, I thought you were talking about the things the fish are alive.
I don't know why we ended up with a fish.
Well, I was looking at the
No.
picture.
I'm just teasing.
Must have caught a shadow there.
Urchin. 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 Fish. Shells.
Is that like a small noddle shell?
No, it's a coral. Over on the side again.
You don't want to look at it, no? What is that? That's a coral?
I thought so.
Why does it have a shell? Is that just all the internal chambers of the shell?
Yeah, I don't know. Maybe.
Would you like to look at it or not?
Nah.
Last time we had mentioned Europe.
So for anyone that was interested in the halometer scoops that we got yesterday, we sieved the water at the bottom of the box that was holding it, and we also put all of the halometer leaves into trays, and were able to extract a number of very interesting microgastropods from that sample. So I'm super excited. There was a beautiful heterobranch, which is kind of like part of the group that becomes sea slugs, but they still have shells. So there are a couple of those. One, I've only seen probably 3 different species of that genus before, so I'm super excited to get one of those out of it. But yeah, it was a real little treasure box, so I'm really glad we're able to do that. Steve? Not sure. Yeah. It's a fun game.
Leo, John, you can come down another 10 meters, please. 150 wire out.
A lot of ridges off to my right.
Yeah. Let's go and see. Sand ripples? Is that? Yeah. Wow. Look at these. If I were a Milo, I'd hang out here.
A lot of logs and sand.
See more sediments.
Yeah.
I love sand waves. They're so gorgeous.
Little runs. Leo, John, come down to 155 wire out, please.
155.
Look at the sonar.
It's awesome.
It's cool, huh?
I'll copy Leo. If you just stand by there, we're just going to reset up here. Reset up, so about to go. 2 seconds.
We'll see if anything gathered down in here. Anything small, or are you just looking for the big stuff?
I think I'm holding out everything for Milo.
Okay. If I can fly a little bit higher, then the quicker. Because they're gonna be bigger than
Oh, yeah.
that urchin, right?
At least, I mean, that size or bigger. So yeah, we won't accidentally go past one.
Leo, John.
What do they feed on there?
He said his name was.
Other mollusks.
You can bring her back up to the surface, mate, and 30 meters per minute, please.
Yeah. That's why I was like, oh.
Hectic. Mate, it's hectic. Fair dink. What was the one that Andy was last name?
Struth.
Stop.
Fixing,
Yeah, fixing it.
That's not one of them, huh?
Finnegan, right?
More of a resemblance to something else.
Or imagine.
I've seen you before.
Imagine.
You have a little cave.
Oh, what a cute little hidey hole. No wonder it doesn't go anywhere. Come in. Oh no, it's the
Out on the shelf.
Yeah, it's alright.
Yeah, now I'm going north. Yeah,
Yes, we are.
I'm crossing my track line right now.
Yes. Well, at least you know where you're going. That makes me feel better. This is the kind of landscape that gets me really lost. Oh, it's only 21 degrees now.
Tiny voltage variations. You put this one a bit short out on the other line.
I think Carl did that.
Doesn't make sense.
Hole in the ground. Meteorite. Is it going to be more sandy stuff?
Yes.
Oh, Jason?
Cody, just to reconfirm, yes, what's the CTD on deck? We've got those now. You're still planning for 1700 roughly?
Once, or we can wait till the CTD comes on deck?
Yes, yeah, that's correct.
Yeah, because once the CTD's coming up now, so 50 meters.
Sounds good, bro.
Tell him that you're sitting beside the CP.
Finishing the day as we started it, flying over sand.
Yes.
What's that?
Finishing the day as we started it, flying over sand.
General call. ROV recovery in about 10 minutes. 10 minutes. Bridge, they recover about 10 minutes.
Why is it going to be upset?
It's not necessary to show up. That's a big crowd.
Right, Joey, ROV recovery in 10 minutes. Where is Phil's going to be on the bridge? Thank you. Phil on the bridge. Thank you.
Fiddler on the bridge?
Yeah, fiddle on the bridge, fiddle on the bridge, fiddle on the bridge, you in the fridge, fiddle on the bridge, and you in the fridge. That's one more life.
Keep it together, come on.
I think we've been on this boat for too long now.
Yes. Should have seen us after 5 months.
Possibly glad I
Honestly,
didn't.
yes. Honestly, I'd rather not.
I'll copy Leo. You mind bringing it up so that the power sensor is out of the water for a moment? Sorry, that's just the white ball. If you bring it up half a meter.
A lot of those little caves. that one?
No.
Leo,
but
could you come up with an
they
half
might
acre,
have a
please?
shape like that. The long. Oh, it's dead. That might be the shell though.
Yeah, just up slightly more so that the top is sticking out of the water.
Yeah. Oh my god. Oh my god.
What, is this it?
Well, it's a dead one. Yeah, that's what they look like.
Do you want to have a look at the shell?
Okay, that's awesome.
Probably just make me sad. I think we should zoom around and keep looking.
So it's going to look that shape, that size,
Yeah.
or is that broken up a little bit?
It's broken a little bit, but it's probably not a giant one. That is it. Oh. Far out, that's the closest we've ever been. Oh, yeah. It's okay.
That's too invested.
Me too, I didn't think.
What about that one?
Could be the top of one of the shells. Oh no, it might be a different shell.
Big old hermit crab?
Yeah, that's it. Yeah, that's a different shell.
It's a pretty big hermit crab.
Yeah, it is. Well, we won't steal his shell.
Big swans.
Yeah, big. Big swamp.
You name them Tiny? Oh, that urchin. Oh, I'm gonna jump it. Oh,
Ready,
I'm gonna
ready,
jump
ready,
it.
ready, 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 ready.
Whoops, bro. Do a method grab. The arm could go down, just grab the side.
And landing on this one.
Whoops.
That's what we need to do is look on that spring.
Right. A little bit smaller. Right.
That's a big deal then.
Alright, that.
Alright.
Looks like just in case. See that big shell John picked up. Leaving it in your typical hands.
Roger that, Cody.
Okay, I'll talk to you.
Curious, yeah. You're just leaving them nipped out in case we find one in this last few minutes that we are searching.
Just give me a reminder of how to turn it on. Did you get that in the knowledge shot?
The photo of it?
Actually.
No. Sorry.
Ah, never mind, people. There always needs to be a unicorn left out there. So
We found a shell.
we found the shell of
One
a
step
Milo,
closer.
which is one step closer to finding a live Milo, which is one of the reasons that I'm here on this trip, and it would be amazing, but it hasn't happened today, folks, sadly. So we have to wrap up this dive now. Thank you so much for joining us again on the Twilight Corals expedition. This is the end of dive number 417, and we will be back tomorrow to, unfortunately, have what I think is the last dive of the expedition. So hope you can make it. Stay safe out
A
there.
very special event tomorrow.
We do, and that very